Did you know that mycology is one of the few scientific fields where amateurs play a pivotal role? Local amateur mycologists often have extensive local knowledge and keen observational skills, enabling them to notice subtle changes in fungal populations and habitats. Many amateurs are driven by a passion for fungi, enjoying the challenge of identification and contributing to a greater understanding of fungal biodiversity. Many new fungal species are discovered and documented by amateurs who often explore under researched or remote areas. From urban parks to remote forests, dedicated hobbyists are out there collecting, identifying, and even discovering new fungal species. Their passionate involvement significantly enriches scientific research and public appreciation of fungi. In this episode, we will explore how these passionate amateurs are revolutionizing our understanding of fungi through advancements in DNA sequencing. We'll uncover the unique blend of accessibility and collaboration that defines mycology, where citizen scientists contribute valuable data, participate in groundbreaking research, and champion fungal conservation. Join us as we reveal the powerful impact of these unsung heroes and how their work shapes the future of mycology. Get ready to be amazed by the hidden world beneath your feet and the amateur mycologists who bring it to light. That's right, I'm talking about Kyle Canan and his team at the Ohio Mushroom DNA Lab.
You're listening to the MycoGeeky Podcast, a podcast that inspires people to grow mushrooms at home to improve their mental, emotional, and physical health. Most people call him geeky, and he is a passionate mushroom cultivator, advocate, and educator. Every week, he sits down with fellow cultivators, mushroom educators, scientists, and therapists to discuss the various ways people can approach mushroom cultivation and how mushrooms can be used to improve their lives.
All right, what's up, everybody? Welcome to the MycoGeeky Podcast, the podcast that goes deep so you can level up your at-home mushroom cultivation game. I'm your host, MycoGeeky, and we got an amazing show for you tonight. I have been slowly over the last year or so getting to know a couple local guys here, guys and gals, and they are definitely teaching me a thing or two. Uh, I'm talking about first and foremost Kyle Canan. We met at NAMA last year. We talked a little bit before that, but definitely got to know him at NAMA. Got to know him a little bit better at Ohio Mushroom Festival, and then done a little foraging with him from time to time. Anyway, he's a cool guy. So had to have him on to tell the OMDL story. The Ohio Mushroom DNA Lab, if you don't know, is a passion project of Kyle's. He decided we're going to democratize fungal barcoding. We're going to get people out looking for mushrooms, and we're going to start figuring out what we got. Hugely inspired for sure by Stephen Russell. Um, he who we've had on the show to talk about a little bit about this. So tonight this is going to be the OMDL show. We are going to really get into it with some of the crucial players on the team. And shout out to the people who are not on the show that are part of that team. You guys are doing great work. And every time I log on to my iNaturalist account and I see some results back, it's usually because of those guys. So thank you so much. Before we get into it, though, I want to do quick shout out to Stealthy Spores. Um, it's super fun. If you guys want to get yourselves one of these little hero cards here, a little Myco Geeky hero card, you can get the whole the Halo, whatever it's called. I almost said holotype. Yeah, get the holotype card. Those are the best cards. 10% off if you use the promo code Geeky. All the kickback for that goes to Myco Mama's Mycelium Revolution. So yeah, if you guys want to own a little part of Cubensis cultivation history, it's a pretty cool way to do it. So also want to shout out my Discord. You guys are amazing. Love what's going on in there. We're keeping it pretty low drama. Which, man, if you're spending any time on some of these social media platforms, is not always easy to do. So shout out to my mods. You guys are running a great little system there. I am not popping in there as much as I would like to. I try to get in there, answer a few questions from time to time, but the real thanks goes to all my moderators. So you guys know who you are. Appreciate what you do. Also, Mycoholics Anonymous, I just want to give you guys a little update. Man, I've been catching some flack for holding. I set the bar on that one. I said I really just want this to be an advanced group. I want really advanced ideas to be presented. And man, I'm gonna be real transparent. I haven't really talked to the moderator group about this or anything, but I wanted to bring it up on the show tonight. It doesn't seem to be what people want. People want access. They want to talk to advanced growers. So I think you're going to see that over time we're going to evolve the membership criteria and try to make that be definitely have the tone be advanced, but allow serious amateur like newbies a chance to get in there and soak up some knowledge. So just a little update on that. Stay tuned for more on that as we figure it all out. Also, man, mushrooms are out. Get out in the woods. Take a hike. Go enjoy it, man. I'm telling you, I've been out with my kids a lot, almost every day. We're finding mushrooms. They're learning how to identify trees. It is a beautiful thing. You definitely enrich your life by getting out in nature and learning how to appreciate it. On that note, my buddy Kyle, uh, almost every time I'm around him, he will mention something about this Joan Maloof book. I finally bought it. It's right here. It's called Nature's Temples: The Complete World of Old Growth Forests. Um, this woman founded what's called the Old Growth Forest Network. You can look that up on Google, and you can find where there might be some old growth forests in your area. And boy, oh boy, if you've never been in one, it's a special thing. So go check it out. If you're looking for, hey, I want to go hiking. Geeky keeps bugging me about getting out and hiking. I think I'm gonna go do it. If you're looking for somewhere to go, first step one, see if you got some old growth forest nearby. If not, the state forests, those are always great. Large tracts of state forests are always going to be good for some mushrooms. Huge, tree canopies, that's what you're looking for. And boy, oh boy, if you can find a mushroom anywhere in those woods, but when it starts getting that like light, dank, mildewy tree leaf litter kind of vibe in there, then you can slow your roll and start really paying attention. You will definitely find some mushrooms. Another book that just hit the scene, Kristen Wickert. She has a PhD in, I think it's like plant pathology. She just did a book, The Plants of the Appalachian Trail, Hiker's Guide to Almost. Girl, why didn't you just do 400 species? I mean, that's so specific. 398. Couldn't find 2 more. That's okay. That's a scientist move right there. That's a real scientist move. She said, "Nope, I'm not gonna do 400. I found 398. That's what it's gonna be." This book is, even though it says plants of the Appalachian Trail, it it's got mushrooms. It's got other things. She did the whole trail. Look at this. My brain's going backwards because I can't figure out what thing to move. There we go. She hiked that whole thing. It's like from like Georgia all the way to almost the northern part of Maine. 17 different zones. She's got plants. What else do we got? Trees, plants. I'm looking here. A lot of trees, a lot of plants, a lot of shrubs, lichens, wildflowers, a lot of wildflowers, ferns, mushrooms. What else do we got here? Sorry, you're just watching me come through this. Anyway, this is her right here. You can follow her. That's her. You can follow her on Instagram, K-Dubs, the hiking scientist. She's cool. I've been following her for a while. I always learn something from her posts. She's like another one of those high quality Instagram uh, content creators like Alan Rockefeller. Every time, they do a post, you're going to learn a little something, something. So check that book out if you're interested, especially if you live anywhere near the Appalachian Trail and you're looking for a little book to hang out and bring along. This is a good one. It's chock full of a lot of species information there. So check that out. And man, I'm trying to figure out if I forgot something. I don't think I did. Um, so let me just tell you guys this. I am hugely interested in seeing the OMDL lab just grow and grow and grow and grow and grow. I, of course, selfishly just want a strong faction of mushroom loving people in the state of Ohio where I live. But more importantly than that, I really see this growth that is occurring with this barcoding movement. The New York Times just did an article about all these guys: Stephen Russell, Alan Rockefeller, Mandie Quark. Um, I'm sure I'm forgetting a bunch of other people. I have not read the article yet because it's paywalled, and god dang it, I'm just going to go to Barnes and Noble and buy the magazine, I guess. But um, anyway, it's gaining attention. Um, it is of interest to the population, and I think we are living in a time where there are going to be a increasing number of people who want to get out into the woods, who want to figure some stuff out, who do have a passion for fungi and want to figure out where they can put that energy. We've talked about this a little bit on the show. So that's what this episode's all about tonight. We are going to show you guys step by step how OMDL came to be, how they work, the roles different people play in it. And if you have submitted a sample or several samples to Continental Blitz, and you've gotten some ITS sequences back, and you've gotten your barcode back, you're going to now know exactly all what goes into that entire process, and hopefully gain a deeper appreciation for all the hard work that these guys put into this, for no money, providing a free service to you and me and everybody else. So without further ado, let's get into it. Kyle Canan and the Ohio Mushroom DNA Lab. All right, welcome to the show, Kyle Canan. What is up, dude?
Oh, not much. Same old here in the lab. How are you? Thanks for having me on.
Oh yeah, man. Um, yeah, in the labs, that is where we tend to be. You are just cranking out the work. Man, cranking out the work. So we just so you guys watch and know, Kyle and I, we get to hang out from time to time. I keep them busy. A few of my other buddies in town here, we definitely keep them busy with a lot of mushrooms. There are a lot of mushrooms in Ohio, despite what Stephen Russell says. We got a lot of mushrooms here. And
That's right.
we're trying to let Indiana know what's up. So we're keeping them busy. And I said, look, man, we got to come on and talk about OMDL. We haven't really just highlighted Ohio Mushroom DNA Lab. I'm an Ohioan. He's an Ohioan. It just had to happen. So here we are. We're doing it. I'm glad to have you. Um, so instead of mushroom origin story, I guess what we need the OMDL origin story. How do you go from regular dude who likes mushrooms to regular dude with a min ion who then stops being a regular dude and starts being a super cool OMDL dude?
Well, if you put it that way, thanks. Yeah, so I was on the podcast before and I kind of talked about a little my history with like finding a new psilocybe. And then I found out, this whole thing exists called DNA barcoding. So that spiraled into, myself obtaining equipment and stuff and try to start doing my own DNA sequences. Um, but that was quickly not enough. So with the Sanger equipment and the price of Sanger, I wasn't able to, afford all of my specimens, let alone some of my friends. So then we get into, yeah, how did we all click up and, rally, and now we're all working on all this DNA work together. So yeah, just once I found the psilocybe, met a guy named Scott. He also found a psilocybe. We all know this by now. And Scott was shared a lot of the same interests with me. He was like, "Well, I really want to get all my stuff DNA sequenced, and let's figure out how we can do this." So Scott was like the first person that I started like taking specimens from, and I was just sangering it up here at home, just extracting and PCRing and sending it in for Sanger and doing like 8 or 16 at a time. But the cost obviously is that factor. So I quickly discovered Nanopore through Stephen Russell, went to one of his talks. My mind was like, I was like, okay, this is crazy. Studied, went to one of Stephen's classes, obtained the Nanopore, obtained all the stuff he needed. So now I was in a predicament where it's like, okay, now I need, a lot of specimens. So along that line, I started meeting, individuals in Ohio and other states. And anytime I would get a sequence back, Scott and I would work on it. We would validate it over Zoom, like live, basically. We'd get all excited. And then we would post those. Like we would post them in Ohio Mushroom Enthusiast, a Florida Mycological page, just, DNA or mushroom enthusiast pages across Facebook. And with that, then we kind of started getting like a lot more attention. Folks are like, oh, this DNA stuff, it's really cool. Like, I want to get my stuff sequenced. How much do you charge? And I really didn't have that answer yet. I can't remember when I made that decision, but I wanted to make DNA sequencing to, the average folks accessible for free. So I started to game plan some ideas, a lot with Scott. Scott and I were like the first two to really just kind of put a lot of things into perspective. And then, yeah, we just started asking folks to start sending in samples from, we really wanted Ohio because like you had mentioned, not that it is a race, but it is kind of cool to have somebody like Stephen Russell and all the work he did to look up to. So I was like, well, let's try to get a lot of Ohio people on board and get these specimens. But what really kind of turned out was it wasn't just Ohio people that were interested. And then when we started saying, hey, we're going to do this for free, people like from every state were like, hey, I want to send mushrooms in. So we started accepting specimens, and along that way, I started meeting, more and more folks. Last year was a big year for me, traveling and going to a lot of these mushroom events. And yeah, one thing just kept leading to another. I would meet some individual, we would click, like the Step Brothers thing, like, did we just become best friends?
What? Did we just become best friends? Yep.
You like DNA? This is crazy. And so we just started bringing people on to what we call now a team. And we brought folks in from Ohio, Sarah Culliton, Jessica Williams. They were our first two people addition from Scott. And shout out to them really quick. They kind of caught my attention very quickly in those early days when we were making the post saying, hey, can we get specimens? Stephen Russell has like 10,000 in Indiana. We have a long ways to catch up. Sarah and Jessica, they were about it. They showed up. They sent, hundreds. I would say I stopped counting when both of them sent me 500 specimens.
Yeah.
And then they were like making it a game amongst them and kind of like getting other Ohio people. It was pretty cool. So they came on and a bunch of other people came on. We got Alan Rockefeller on the team, Mandie Quark. And then I also met this cool dude named Zach. And I'll let Zach kind of share a lot more, but I thought it was interesting when I met Zach because most of the other people that I met that, share this team, they had like an interest in fungi, and we all have that, fungi origin story. But with Zach, when we clicked and met, we actually didn't have that in common, but what we had in common was Nanopore. And I quickly realized that Zach had, he knew a lot of stuff in this game. And I was like, we got along and everything seemed right. So I was like, this is cool. So we brought Zach on, and Zach has a background in bioinformatics. I can let him explain all that. So bioinformatics are super helpful with like the Nanopore pipeline. So he's been able to do a lot with the Nanopore pipeline for OMDL. Essentially, I give credit to Zach for all of the data streaming and pipeline setup. I couldn't have done it without him. So thanks to Zach. So a few other people, we have like Grant, Josh, and everyone kind of just like fills a niche in the team. Obviously, I'm the guy that does most of the sequencing here, but we also have Zach who has an nanopore, and he is doing some of that as well. And we have validators on the team. Those are like Scott, Jessica, Sarah. Scott being the head validator, the lead validator. I really don't like using that term for Scott because Scott does way more for the team and for mycology than validating sequences. Um, but that's kind of where he took over, and I kind of just fell out of it. I was like, well, the you know DNA analysis is another bottleneck essentially, and it takes a lot of time. So with bringing people on to take care of that would free myself up for more time in the lab, doing the actual sequencing and preparing for that. And yeah, just cool things along the way. So as we're sequencing, we're finding these cool results. And what we're also doing with the team is we're trying to take some of those results and turn them into stories, publications, correct some taxonomy, and things like that. So we have together as a team worked on several publications in the year of 2023. One being like inaugural paper for OMDL, a couple others being new descriptions, and then some others just being some really cool stories. So we take pride in that. That's a big part of our focus. Aside from the actual sequencing and validation, is then uncovering that, making it into a story or publication, and then getting it out to the whole mycological community for everyone to see and enjoy. So
So first off, I just want to say this. Y'all could just you could just do the barcodes. You could just pump out ITSs and just give them back to people
yep,
and go, you do whatever you want with them. I don't give a crap. We're a mushroom lab. This is all we got to do. That you guys are also going, but I mean, that's useless. Maybe at some future date could be useful, but if that's all we do, then we're just like a machine. We're doing one thing. But of course, because the founder loves mushrooms and loves taxonomy and wants to know what he finds, you go, well, we can't just do the barcodes. We got to actually validate. We got to actually contextualize all this information somehow. And I think that is the piece that I want to make sure people understand is that while most people go, I just want to get a barcode, I sent Scott a couple cool mushrooms I found and I want to get a barcode back. Why? Well, you are giving everybody the why. I think this is how it gained steam in the beginning was because it was your posts were more useful than just look at these 4 letters in a sequence, right?
right.
Like this is why those letters mean anything. So
Yeah.
I think that is super cool. I also really yeah, I also saw Stephen Russell give his little nanopore talk and I was just like nanopore
Yeah,
what I
it's mind
couldn't.
boggling. Yeah,
It is crazy. I'm going, yo, when they did the human genome, like if they had Nanopore back then.
yeah,
You
no,
know,
definitely, yeah.
a million base pair reads what? Like all he just kept saying stuff and I just kept going, wait, what? What? So as the software gets better, they can go reanalyze the reads and maybe improve upon them. What? Like I could not believe how cool this was, but at the same time, newer technology, not as many people using it, not as cut and dry. And so when he was saying, "I'm just doing it, I'm just buying these flongals, I'm just trying to get all this figured out," I was really like, "Man, hats off to you, dude, because that is a big project." And then when you stepped up to the plate, the first thing that popped into my head, there is this thing on YouTube. If you go to YouTube, search first follower theory. It's something like first follower theory. Anyway, it's this video of this concert and this guy's just crazy dancing and everybody's just looking at him like he's a weirdo until one other guy gets up and he starts dancing with that guy. And now they're having a good time. They're partying. Now a couple other people start dancing. And by the end of this like 5-minute video, it's like 180 people are all dancing. And the whole point is it isn't the first guy that makes it a thing. It's the first follower. It's the first guy that goes, I like what you're doing. I want to do that too. That sets a tone. And I said, dude, because Kyle did this, this is going to be the beginning of this amateur mycology movement. I promise you in 5 years, there are going to be even more people doing Nanopore. You guys are all going to be working together. I'm super excited about all that. And then when you told me about Zach, I was like, oh, and Zach's over here doing all this crap with Nanopore, and now he's joining the party. Yes, 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 yes. This is cool stuff, man. I am so excited. It's been amazing to watch you do all this stuff, assemble the team, figure it all out, make it, a more well-oiled machine. It's really exciting. And I can tell everybody watching, I sent them, man, Sarah and Jessica made me feel weak as all get out. I sent like, well over 100 and some specimens. I thought, yeah, it's a lot of specimens. And then Sarah's like, yeah, I think I sent them 545. And I'm like, okay, I tried. All right.
Yeah.
I give
Yeah,
up.
they're on a different level with it, and that's
Oh,
respect.
yeah.
That
Yes.
they can have it. It's not for everybody, that's for sure.
Yes.
But yeah, you definitely provided quite the amount of specimens yourself. Not only just Ohio specimens, you also contributed
Oh,
from Mexico,
yeah.
which we
Yep.
got some.
Nama, Mexico. Yep.
Yeah.
Yeah, that's fun. Now, I'll tell you what I think's really cool is right the proximity. I don't know if I've heard anybody talk about this, but Steven's got Indiana,
Mhm.
and it's pretty cool that you're in Ohio because now you got neighboring states, so now you can compare how similar are all these things, right? These things
Yeah.
that we think are the same, are they molecularly the same? Over time, as the years go on, do they stay the same, right? Oh, you got some bolete that you sequenced 2 years ago, you sequenced it again, and it's from the same spot by the same guy. And now it's a base pair or two off. I think there's a lot of interesting things that will start happening over time by having such an ambitious project, building these data sets, guys like Zach helping to figure out what to do with all this data. I think it's really cool, man. So I'm glad to have you on. I'm glad we got a chance to kind of paint that picture. Let's bring the team on, man.
All right, let's do it.
Let's do this. Let's start out with Scott. When you said, it was like in Step Brothers.
Did we just become best friends? Yep.
Yeah.
I'm like, oh my God, you found a novel species? Oh my God, I found a novel species.
Yeah, we
Yes.
kind of call each other the Philosophy
Yes.
brothers because
The Philosophy Brothers. I
we
love
on
it.
that,
Yes.
and it was just rolled
Yes.
one after another.
All right, man, let's bring them on.
All right.
All right, what's up, Scott?
Not much. How are you?
Man, just living the American dream in my basement, hanging out with some fellow mushroom nerds. It's a good night. Yes, yes, yes.
Oh,
So,
yeah,
man,
I'm happy to be here.
you guys, just what do you do? We just wander around the woods looking for mushrooms, and you had me at I discovered a psilocybe. Why don't you both talk a little bit? I mean, the stories I know Scott talked about a little bit, but just real briefly for people who might not be familiar, why don't you both just talk quickly, tell the story about your psilocybe finds?
Go, Scott.
Yeah. So actually, the first observation of Nivea tropicalis was back in April of 2016 by someone with the handle the Grim Raper on the Shroomery. But they never retained the collection, and the photos were not high quality. So it remains just like an unverified observation of it. But then in 2019, April of 2019, me and my buddy Andrew Chambers found the same mushrooms growing in front of the security gate in front of a gated neighborhood in mulch. in Palm Beach County, Florida. And at the time, I was familiar with the various species of psilocybe that were known to grow in South Florida. And this one didn't really fit the criteria for any of them. And, I went over this on the last episode I was on, but, eventually I got it DNA sequenced. I found out that it was most closely related to various species in Thailand and India, namely psilocybe thairugoneomaculans, psilocybe thaiduplicatus fastidiata, and psilocybe weyanodensis. And after doing microscopy and additional sequencing of other gene regions, including LSU, RPB1, RPB2, and TEF1, and obviously literature review, I determined that it was a novel species. And since the last time I was on, myself and a couple of co-authors have officially published this as an official taxon in the journal Maclovania.
Very cool, man.
Yeah.
Kudos, dude. So here's the deal. I wonder how many people have found a new species of mushroom and even thought it was a new species of mushroom. And that's it. And that's as far as it went. Like you said,
Yeah.
your dude 3 years prior who, I feel bad for his screen name, but he hops on there and low res pictures. I don't know, 2016. I feel like he could have taken a good picture. I don't know. Maybe he had like a Nikon cool pics and that was as good as he could get. Yeah, I'm sitting back going, how many people are just walking by novel species? How many people are
Right.
or not even novel species, but just something pretty freaking rare? If you don't take the time to look at it and think about it, you never know. And you did the complete opposite of that. You went all the way to the finish line with it. And I think that's cool. And people don't have to do all that. They can pass the baton too. They can go, here it is. I know these guys, they know how to do this. I'm gonna send it over to them. So yeah, man, that's great. Now, okay, so Kyle, same thing. Let's get your psilocybe story in.
Cool. Yeah. Well, first off, congratulations to Scott and the Nivea tropicalis being published. So Suliryza, some similar story to that. I also was not the first person to observe it. It was observed for the first time in 2021. That's when my observation was. It first popped up, I want to say, in Pennsylvania. And then shortly after that, popped up in Iowa. They were well documented. One was on Mushroom Observer, the other one was on iNaturalist. And then I found mine, and I think shortly after that, it was found in Indiana by another user. So Ross is the user in Iowa that observed it, and then Eric Hensley observed it in Indiana. I'm not sure the person that observed it in Indiana. So I found mine in Ohio. I was with my friend Doug, and we were on a winter tree ID walk. So it was, it's December. It's like 40 degrees. Didn't expect to find any mushrooms. And at the end of the walk, we just like looked down, and I'm like, dude, Psilocybe ovoidiocystaediana, and that was my first because that's a species that is found all over the Ohio River Valley Basin, and we were Southern Ohio, and but that was just like a quick guess, I was like, this is weird, like December, and as my heart started to, my heart rate started to calm down, I continued to think a little bit more in depth of this, and I was like, this is interesting. So bring it home, run micro on it real quickly, get the spore shape, realize it's probably not an ovoid. Got it off to Alan, and simultaneously while Alan was working on it, Stephen Russell was also working on the collection from Indiana. I want to say the sequences kind of came back around the same time. And yeah, just I got the sequences, did the legwork like Scott was saying, do the micro, you get the DNA, you do some analysis, read the literature, and yeah, quickly found that this also was a new psilocybe blooming mushroom, and it has a winter phenomenon. All of the observations across the Midwest and the East Coast have only been found between, I want to say, November and January, but most of the observations being like December. And we have
a winter psilocybe.
Yeah, the common name is going to be winter teachers, and the paper's complete and should be published this week. Thanks to all the people that helped me with that paper, Scott being the number one guy that helped, and we're publishing in the same journal. So on top of our whole free sequencing movement, we also just believe in free access to everything. So we've chose to post all of our publications so far in a free journal, where people can obtain that information just as easy as we could obtain it.
Right, that's great.
So that's the game plan. So it should be published, well, within this month, I would say.
Love it. Dude, I didn't know it was a winter fruiting.
Yeah,
No way.
Both times I found it in 2021 and then again in 2023, dead of winter, cold, no leaves, frigid. Not in snow though, haven't we? None of the observations that I've seen yet are in the snow, but definitely cold temps.
Dang. Right, now I got something to go look for in the winter.
There is an observation up in Northeast Ohio that's, you can say it's unconfirmed. Maybe. I believe it is. Yeah.
All right. I got a new task for the winter. I love it. Yeah. Usually I just shut that part of my brain off in the winter and I sit around and Jessica and Sarah and we just fiend for the spring and make fun of people who take blurry photos and can't figure out why INAT can't figure out what it is.
Yeah.
I would much rather be in the woods.
Yeah, no kidding. I encourage everybody to go out in the winter, because I don't believe that would be like maybe the only philosophy, but I believe that there might be other winter mushrooms, that we are missing.
Dude, I mean, it's just logical. The only people out wandering around the woods in the winter, I don't know, maybe some hunters or like it's just people aren't wandering around the woods in the winter. So how you gonna know what's there? Unless maybe you're just wandering around your backyard woods a little bit or something. So yeah, I got a new task. I'm gonna have to start snowshoeing up here. That'll be my new rabbit hole I can go down, as competitive snowshoe mushroom foraging.
Yeah, and one thing I didn't mention, also found in mulch, and all the observations are found in mulch beds.
Oh, in mulch beds. Oh, no way. Okay. Yeah. Okay. So I don't have to go in the woods. Even better.
Yeah. Although it would be interesting to find it in woods. Yeah.
Man, all these mulch bed mushrooms are just fascinating, especially when the phylogeny comes back and it's like, where is it? It's from the third highest peak in Nepal. And you're like, how is that what it's related to? That's craziness. Because we're not getting our mulch from Thailand. So wow, how did that happen? It's crazy, man. I don't know.
We may never know.
Always is that way though. Somehow it always seems to be that way. Random mushroom. Where's it from? Russia, Thailand, Antarctica. What? It's craziness. Maybe not Antarctica. Maybe no Nanopore is going to be set up in Antarctica anytime soon. All the water's frozen there. No good. All right. So you guys just clicked because you were about the same stuff. And man, it's like, so I got a background in music, and I love talking to people about how all the eras of music usually evolve from technology, right? Like, right, whether you're talking about hip hop, back in the day was just somebody taking a turntable and subverting the use of it and doing break beats, whether it's techno, EDM, but all the musical movements are usually generated from some new technology. And so same here. I really see this Nanopore movement as a significant, and when Zach comes on, I'm sure he'll have something to say about this too, but it's just so different, right? It's not like how precious everybody is with their sangers. right? It's like, oh, I got my 8 sangers. I'm going to submit it to that guy in Spain and I'm going to get my 8 precious sequences back. Bro, you're running like 1,000 at a time. It's it's nuts. It's so much more. So let's bring the final piece of the puzzle on, and then we can really start talking about the process here. So welcome to the show, Zach. Now, Zach, how do you say your last name? Geurin?
Yep, you got it, and
Geurin,
you
cool.
spelled it right
Okay,
too. I was impressed when you got that right on the first try.
that was sheer luck. Sheer luck.
Nice.
Yes.
What's up, guys?
Cool, man.
Yeah.
Well, welcome to the show. I have only met Zach once, and it wasn't for very long. We had a beer and we got to chit chat here in Akron a little bit, which was great. But I have not talked to you very much, and I honestly don't fully. I've heard little bits from Kyle about what you've been bringing to the table and what you're all about with OMDL. Why don't you talk about how OMDL hit your radar, why you wanted to get involved, all that good stuff?
Yeah, for sure. Well, like you guys have said, I'm a big fan of Nanopore, and have actually been a user of Nanopore since the very early, like the first days of back in about 2014 or so. I was like fresh out of college. I have a degree in, I got my degree in microbiology and genetics. So I've been a big nerd around that kind of stuff. But I was in the bacterial world doing sequencing bacterial genomes, um, and working on Nanopore and some other sequencing platforms. At that point, it was in like a beta access program. You had to apply to get into it, but it was still so incredible because it was $1,000, and the other sequencers I was using were $250,000. And so when Nanopore hit the scene, it was like a pretty big deal, even for um, even considering its flaws, and it gets flack for its error rate and stuff like that, and that certainly had merit. But um, yeah, I was a fan from the beginning, and watching the development over the last decade for the advancements it's made, and uh, in the kind of technical capabilities of it and all that stuff. Um, I've just always been a really big nerd for that kind of thing. And then I was introduced to Kyle early last year, um, yeah, through a mutual contact, and heard about what he's doing. And at the time, he wasn't quite like hands on with Nanopore yet. I think he had taken Steven's class, but hadn't done a run himself. And so we met briefly, and I was like really impressed, super impressed with just his passion and really going for it, starting his own lab and everything. And we kind of just left it like, I was like, hey, let me know when you're ready to get going. um and then a couple months later, he reached out and was like, hey, I'm about ready to do my first Nanopore run. And I'm like, yeah, let's go, let's do this. And from that point on, I've been kind of serving as like the just overall like tech support. When it comes to Linux, a lot of stuff in bioinformatics happens in Linux.
Oh, I remember there was one day Kyle on Facebook was like, this mother beeping Linux is gonna be, I'm
Yeah.
gonna throw it out the window. And then like, I don't know, a day or two later, somebody solved the problem. I don't know if it was you or Steven, but
It was
I
Zach. It was Zach.
I figured.
Oh yeah, Kyle and I have been on many long and late Zoom calls where we're remoting in to help him figure it out. But it's obviously all worth it, in the end. Like, we got him up and running, and like you said, he's really just taken off, especially recently, just like cranking out run after run, tearing through samples. But yeah, I guess like I said, I was always in the bacterial world. I worked with like food safety pathogens and a lot of clinical pathogen work and stuff like that. And I've certainly always been a fan of mushrooms, but only really jumped into it as of a couple years ago. Started really looking into it. So it's both been familiar in the next gen sequencing aspect and all that, but also like so new to me, learning, the intricacies of ITS barcoding and stuff.
So yeah, our bacteria for identification purposes, it's not ITS, is it something different for
It's
bacteria?
different. It's similar in idea and in the concept. They use a 16S gene. It's also a ribosomal gene. Uh, and I had done exactly that for bacteria and actually for fungi, but using Sanger. And I worked in a cannabis lab for a couple of years, so it's kind of funny. I was doing fungal ITS identification, but it was for plant pathogens, and figuring out why people were failing their yeast and mold tests when they were trying to pass quality tests for cannabis production. Um, so I had some idea of it, and I've also done a lot of environmental sequencing stuff, like they call it metagenomics, where you take soil or water and just sequence everything in it and sort out like all sorts of this mixed life, a community of life that's in it.
Which gets way easier with Nanopore compared to anything else, right? Like,
Yeah, I mean,
what do
yeah,
you do
it's
with a soil? You can't send Illumina a soil sample, right? You would have to do a monstrous amount of prep
you
on that.
technically could. The real advantage of Nanopore
Oh,
though is the read length. Like in from its
okay.
very inception, I mean there's a comparable platform, PacBio, that also does longer reads. That's the key thing of it. But PacBio is like $1 million, and you pretty much have to hire like a full-time engineer to maintain the instrument. So yeah, certainly like Nanopore offering that for $1,000 up front, um, and yeah, basically the longer read you can sequence, the better resolution you have overall. You can really drill down to like a species level. Like actually, it's a great example. One of my metagenomics projects on Illumina, with bacteria, uh, we were hoping for like genus level at best, maybe even like happy with a family level identification, if you're familiar with like the taxonomic hierarchy of how that goes down, drills down. So yeah, with Nanopore, being able to get a conclusive species ID with most of your sequences, that's huge. And then doing it for a cheaper cost, obviously making it accessible for citizen scientists and amateur scientists. It's That was always really cool, and I was like, that was one of the things I always loved about Kyle's mission, is just his commitment to doing it for free, and everything open. I've been a big computer nerd my whole life, and I'm a big fan of like
It's
open
way
source.
like Linux. Yeah.
Yeah, yeah. Open source software is a beautiful thing. I love like the idea of everyone contributing for the benefit that everyone can enjoy, and so like all aspects of it. Really, I was like, this is perfect. I would love to help out with this, and um, yeah. I mean, now it's we realize we're right about at the 1 year mark from when Kyle had done his first run, and we've published what was it, like 3 or 4 thousand sequences to GenBank now, which is like the big public repository of all sorts of genetic data, but in particular, like when we get our final validated ITS sequences, that's kind of like one of the main hubs that we can upload it to.
Yeah, we're just under 3K right now on
So
GenBank. We have 3 runs in queue to be uploaded to GenBank, so another 1,800 or so.
All right. So 1 year, 3,000 sequences in GenBank, 3 papers, 1 year.
yeah, and we also we published a paper, and it's kind of funny how quickly we surpassed what we were able to put in, like the data we were able to put in that first paper. It covered our first 5 runs, um, and since then we've more than doubled what was captured in that first paper. So again, like just really cranking out the number of samples, and having people like you and like Sarah who can send in hundreds of them at a time. Like it's pretty cool to actually be able to offer that to people. They just go out, find a mushroom. I'm again somewhat newer to it, but like my family lives in northern Michigan, some like really prime mushroom hunting up there, and I've been out several times with them and getting them involved with like finding and recording the observations and all that stuff.
Got to train them.
Yeah, exactly.
Yes.
You got to get the right gill shot, you know, all that stuff.
You got to get them trained. Now, are you talking in the mitten, or are you talking Upper Peninsula?
In the mitten, yeah, not proper Upper Peninsula, although I do enjoy the Upper Peninsula quite a lot.
So how far up there are we talking about?
Traverse City area.
Oh yeah, that's where I was. I was born in Traverse City. Man, anything north of Cadillac is like God's country up there, dude. It's great. Yeah,
I was born and raised in Michigan for 30 years before I moved here to Chicago. But oh man, going up north in Michigan, that's something special.
Bro, those pure Michigan commercials, dude. When I lived in Southern California, I had a couple people be like, "Yeah, I've seen those pure Michigan commercials. Those are bullshit, right?" And I'd be like, "No, bro, those don't even do it justice. I'm telling you."
Yeah.
Like, in the winter, everybody wants to kill themselves. In the summer, though, it's pretty phenomenal. Yeah.
Yeah.
That's cool. You're getting the Michiganders trained. I love it.
Yep.
We're gonna have the Midwest. It's just gonna keep going. It's just gonna keep going, and then hopefully we can move down south. Yes, maybe Steve goes one direction, Kyle goes the other direction, and then Scott comes from the bottom. And pretty soon, 10 years later, we got it all over the place. I think that'll be very cool. And man, right now it's so new, both the nanopore technology, but then also just the amount of data, right?
Mm-hmm.
In 10 years, AI plus all these data sets equals figuring a lot of shit out.
Yeah, that's true. One thing I really liked about bioinformatics is just the idea of like with next gen sequencing, when it came on the scene, it was a new problem of having too much data. Like you literally, it's not humanly possible to scrape through it all, parse through it all. And so bioinformatics, aims to like solve that problem. That's some of what I helped Kyle with is like designing those software pipelines so that you can process hundreds of samples in every run without having to there's still quite a lot of work in the sequence validation on an individual sample basis, but ideally you want to be able to churn out the highest quality results so that it makes the validators work a lot easier. They're not like going through so much bad quality sequences and trash and stuff like that. And we were on a call today, and we are typically on pretty long calls where we brainstorm about like where things can be improved, because we're certainly not at like the perfect solution yet, and there's still like a lot of room for an improvement, but that's kind of the exciting challenge.
Now, so I was talking to Kyle recently, and he was talking to me about, the fungals, and sometimes the fungals work great, and sometimes they don't. And I remember back in the day, I think this was a little bit before Kyle bought one. I remember people were giving Stephen Russell some crap because some guy was going on and on about error rate and accuracy and blah blah blah. And Steven's like, yo, 40% accuracy on a run of almost 1,000 samples for the price point still isn't terrible. Now, of course, this other guy's, he's thinking about it from like an academic scientist that says, well, 40%, that's garbage. That's, we got to use Illumina. We got to use this. But man, I tell you what, the more I hear about this and the more I've been reading up on Oxford Nanopore, it's really a democratizing product. To at its price point, like I
there's
can't go
nothing like
buying there's nothing like it.
I think
like
they
$250 million or $1,000.
Yeah,
and dude, you just said $1 million for Pack Bio, and then you got to hire a full-time engineer who's got to have a freaking PhD. Like, only certain people get to play that game, and they're not people, they're companies, and they're really well funded.
Yeah, like I said, Nanopore does have like there is truth to some of the criticism, and it has made strides, especially in the last 5 years or so. they have yearly conferences where they talk about what they're working on behind the scenes, and it's, they put it on like a Apple conference, like tech show or whatever. And they're continuously making improvements. But it does come down to the air quality and what can you do mostly on the data side to counteract that and compensate for that sporadic air quality. And the answer is actually mostly in what's called like a consensus sequence. And so if you think about like a power by numbers scenario, maybe an individual read has some sporadic quality problems. You're going over the AC, G's, and T's, and every once in a while you have like a really bad quality base. Well, that can kind of be corrected if you have a matching read of the same genes region that has a higher quality. Then you can just substitute in like the higher quality base. And actually what happens is if you have enough of that, like 50 or 100 or even more, covering that region, then you just pile all of those reads up and then find like what's the consensus of all of those put together. And the result is you can get very high quality, like very comparable to other sequencing platforms. And that's kind of the idea behind it.
Now, so Kyle talked to me about this one time, and this I had not realized this. In my head, I'm just going, sure, yeah, primers for ITS, hit the beginning, the end, grab a certain little region of some gene information. But with Nanopore, there are primers for everything, right? So there are almost 1,000 primers. Is that right? Where like there has to be tags.
Yeah. Oh, yeah. So you're talking about that's it's not necessarily unique to Nanopore, but it's certainly like suited well for it. The idea that it's called multiplexing, where you can mix hundreds of samples into a single sequencing run, and yet still be able to demultiplex and parse those out and identify like what reads came because yeah. So on the beginning, so you have your normal ITS primers that you would use in like Sanger sequencing. But if you're, for example, doing Sanger sequencing and your specimen isn't pure, if you have contamination in there or something, you're going to get it's going to muddy it all up, and it'll be too noisy sometimes to get a clear reading of what the gene sequence is. But if you attach on the front and the back some unique identifiers in the form of like just arbitrary bases, you add like 12 bases of a unique sequence on the front and 12 on the end, then in software you can basically look for those like an identifying key. And then you have a sample sheet where you have all of your 500 samples laid out, and you're like, these indexes go to this sample. It all works, and then you can split it all back out, and that's a absolutely crucial factor that enables you to get down to the cost per sample being less than $3, or even less than $1, depending on how many samples you can cram into one run.
Right. So now, so I understand that, but I'm still not quite getting the compiling aspect of it. So if, for example, you're throwing 960 samples of extracted, isolated DNA from 960 samples, is each pore gonna grab one of those primers? Is it a little more arbitrary how that works? Does it just sort of find a hole and then run through that hole until it runs out of a strand?
Pretty
And
much.
then but then now can another one come in later, or once one
Yes.
strand? Oh, okay, that's what I didn't.
That's something you pay attention to when you're actually doing a run, because you get a live readout of like your pore count, your pore health. It'll tell you the different categories of like, you will actually be able to see, okay, this pore is clogged. There's a DNA molecule stuck halfway through this pore. This pore has just been damaged or is completely offline, but you have this pore is still healthy. The way it kind of works is those pores will sequence like any DNA that has the right adapter on it. You go through this process after the normal PCR, which is just standard PCR that you would kind of do for ITS, like if you were doing Sanger sequencing. But again, they have those unique identifiers on them so that you have your whole plate of samples. It's safe to then mix them all together because you're not worried about cross contamination at that point. They've been barcoded or indexed. And then you go through another whole process of like the special nanopore library prep kit. The library is just like once the DNA is prepared for sequencing, they call it a library. Part of that is like attaching a little motor protein, an adapter sequence to all of your DNA. And that's kind of the magic. It's kind of like a chaperone that leads the DNA to the pore. And then the pore is a motor enzyme that
because it like has an affinity to that pore.
Yeah, exactly.
Yeah,
But
okay.
if you think about it in like a high level concept, the pores don't really care what it's sequencing, and it certainly they will like one pore can sequence many hundreds of reads, potentially until, like I said, it's something you monitor as the run progresses. You'll see your pore count go down and down and down. There are certain things you can do to really optimize that, like especially with flongals, if you accidentally overload your sample into the flongal, that's a surefire way to actually damage those pores are very delicate, and if you tank your pore count, then you're kind of really hamstringing your run because you've done a lot of irreversible damage. And as you mentioned, the flongals are already kind of notorious for spotty quality control, which
Dude,
is really
yeah,
the,
like what was that, a month ago? Kyle shows up. We're down in South Ohio, and he's like, "Well, when I left home, I had what was it, 70 pores or something like that." And he was like, "Man, we got to get this run going before I run out of pores because it just seems to be going down and down." But then you gained pores right during the run, like some
sometimes
pores
those
woke
clogged
up.
pores, yeah, can be reversed. And not to get too into the technical weeds of it, but the flongals are the like smaller flow cells, as they're called, that has like the membrane pores on them, the nanopores. But there's like the full size flow cell that is like the big brother, or actually the original flow cell, and that is like I don't even know how the math works out, but like 5 or 10 flongals put together, something like that, it equals like a full size flow cell. And with those, it's really cool. You can reuse them. There's actually a way to wash them and reuse them. Kyle and I experimented with that. The big, yeah. Unfortunately, the flongals are meant to be disposable. That's probably in Oxford Nanopore's by design. But yeah, like you could wash them and you will see the pore counts go back up. Like I said, if they're clogged or something, sometimes that molecule will break free, and then you recover that pore, and then it'll start sequencing again.
Now, how long would that large flow cell last? Like,
That's a good question. Something we were also just kind of talking about, like,
because the math might work out where, screw it, man. If you can wash this sucker, because I don't know how many cells are in a large one, but if it's like 5,000, that's a lot of samples. But if you can clean the thing,
yeah, there's
then
something
then you can run it when you feel like it. You don't gotta feel like you gotta, maximize the potential of that cell too.
Yeah, there's with nanopore especially, there's a lot of like theoretical on paper math that doesn't always equal one to one like real world performance and stuff. Flongals is a good example. Like technically you could have 120 pores on a flongal, but realistically we've only seen 100 like once ever, and most oftentimes it's like 70 or 80 is a good one. The full size is I think a max of like 2,000 pores, and you usually see over 1,000, so it is about a 10x, at least a 10x increase from the flongal. And what that means is like, yeah, if you had enough samples, you could, in enough primers that were uniquely tagged, you could run 10,000 samples on a full-size flow cell. That would be a Herculean task probably to amass that many samples and the logistics of it all. But we were just kind of brainstorming or like at least figuring out like, is this something we could or should work towards? And then, like you said, that's just one run. Then if you could
Right.
wash and reuse that, you would have to use like a new primer set. There is a little bit of like carryover, like bleed over.
Hey man, sometimes doing the dishes is the way to go.
Yeah, maybe one day. Like I said, Oxford Nanopore, the company, does their big conferences, and they just got done with their London calling, their UK-based company. And their hot new thing is like the next iteration of the MinION. It's called the Mark 1D. It's even smaller, and actually you can run it off of an iPad now. They have you can still use it like the normal one, plug it into a computer, but it's even more portable now because if you have one of the newer iPads with the latest chips and processors in them to run it, it's just another step in terms of like
Oh,
portability.
maybe it would be faster. Like, does it also crunch?
Not
It would.
necessarily faster. It's more
Okay.
just like the portability aspect of it. I went to one of their conferences in San Francisco a few years ago, and one of the things I was just blown away by is like hearing the stories of how people are using the MinION in the field. I mean, Kyle has taken his lab on the road a few times now, but you know, hearing about people taking it out into like the Bering Sea on like a fishing boat and taking water samples and like sequencing viruses out of, you know, this ocean water, and people doing it in the field for outbreak tracing, like during the Ebola outbreak in Africa in like 2014. MinION was like pretty clutch in terms of like real time live epidemiology and like outbreak tracing. So like that also has like always fascinated me.
Dude,
Like the
when Stephen Russell started talking about wanting to do the soil samples and seeing what DNA was in the soil samples versus what mushrooms were found in that area, I was like, oh my God, screw that. Like, you can just dump a bunch of crap in this thing and extract a bunch of DNA and find everything. Yeah, think about all the little living things that might die by the time you get back to your lab and
Mhm.
finally get to do it. Yeah, being like on that boat, like you just said, in the Bering Sea or in the middle of nowhere and just doing it. I mean, bro, Kyle brought he did a nanopore run just in the middle of nowhere down by Hocking Hills.
Mhm.
Showed up and ran some DNA sequences.
Yeah, that's the
It
beauty
was
of it.
cool.
It's still a little tricky because like we're still working with sensitive reagents that need refrigeration or freezer and stuff. You know, Oxford has long been working on a solution to that in terms of like a lyophilize or like basically a dry reagent that wouldn't need refrigeration. As soon as that is ready for prime time, like that will be a big game changer if they ever get it to that point, because then it would solve one of the last like crucial pieces of making this truly like a sequence anywhere, anytime
Right.
platform.
You know, they're sitting back there though, figuring out how to recoup their R&D. So they're
Yeah,
they're they're always making decisions. How do we want to give them this yet, or do we want to recoup this first? Do we want to? So who knows? They could be sitting on that, man. They could have that ready to go right now, but going, we just got to recoup another 3 million R&D, and then we'll launch that.
yeah, they got to
You know,
sell a few thousand more flongles before they're
there
ready to
there
do
you
that.
go. Yes, yes. All right, well that's cool. And I mean, all I gotta say is y'all. This is like some people like to go hang out at their buddy's house and drink beer
I bought 6 Bush Light, 6 Bud Light, and I love them, tall boys.
and shoot pool. Some people like to go, deer hunting, and some people like to find novel species of mushrooms, sequence thousands and thousands of barcodes, and then do validation on all these mushrooms and, just map fungal biodiversity in different regions. That's all.
Well, you know, going to a buddy's house and drinking beer and nanopore sequencing isn't a bad combination either.
No, yeah,
We did
there
that
you go.
at Alabama Mushroom Fest. It's pretty great.
There you go. There you go. Yes. So you can, that's the ad for Nanopore right there. You can do it drunk. You can you might need to prep your samples sober, but you can do the run drunk. There you go.
You
I
can
like
do it
that.
having a good time. I can contest to that.
Yeah.
Alabama's a good one.
That's
I know
great.
Zach and I've had some good times doing them.
Yeah.
Yeah.
Yeah, man, I still can't get over your Kyle, your posts where you're like, okay, we've done all this thermocycling, all these extractions, and now it's just this tiny little like 0.4 ml tube, and that's got just all 1,000, extracted and multiplied, little PCR reactions in there.
Yeah, like 7 microliters.
Crazy.
And for the audience, that's like 2 drops of water. Like it's
Probably
tiny.
less than that. It's
Yeah,
like that's might be
But
less than like a eyedropper
insane.
drop.
Yeah.
It's crazy small.
Yeah,
very.
people,
Gotta have a steady hand
numbers.
for sure.
What's that?
You gotta have a steady hand for all that pipetting, when you're doing the extremely small volumes.
Extremely small volumes. Yes. One false move and you've just like quadrupled the amount you were supposed to squirt in there. Yes.
Yeah, it might not be for everybody, and like that's also like it's like you're going through this process, and there's so many steps, and it could take you a couple hours, and you don't really know until you're done whether it's gonna work or not.
Right.
Oh yeah. Couple
Yeah.
hours. You're
Oh
in.
man, all I know is I got an uncle who's spent $100 on lottery tickets every week for 30 years, and he's never won more than a few grand, and that doesn't stop him. So look, if you find the thing you like to do, you just keep doing it, right? At the end of the day, they're not all going to be novel species, but that's okay. It's still fun. It's still fun to have some molecular data that confirms, I love it. I love it both when Kyle sends me something, say, hey, ITS is back, then Steven does this to it or Scott does that to it. And it's all, yep, okay, it's a wood blue. I knew that. I already knew that. But it's still cool to get the confirmatory data, or other times where it's like, well, I thought it was this. And then it actually, I might have got the genus right, or sometimes I don't even get the genus right. But whether I was right or I was proven to be wrong, it's still, that whole process is fun. And it's part of the learning process, helps me learn more things, helps me feel good about like, oh, cool. Now I know what's in my yard, or this hike I go on all the time. I know what's around that corner in July. I know it's around that corner in August, and I know what to be on the lookout for. And it's fun. I enjoy it. All right, so can we do this? I have seen recently Kyle has talked about Steven's getting 10,000 samples. You're getting thousands and thousands of samples. I've seen the photos, dude, of of the back of the lab with just boxes stacked. You're opening a box and there's just hundreds of samples in there. I mean, I swear to God, I would need to take a chill pill before opening one of Sarah's boxes. I mean, just you're like, I'm gonna open this box and it's just gonna look like nothing but a whole lot of work.
Yeah.
So you were talking about coming up with ways to streamline that process. So let's take a moment and talk before we kind of tell the full story of how this process works. Let's start with the way you would like to receive samples.
Got it. Okay, and then Scott can chime in a little bit too. I use Scott as a reference because, and I may not plug Scott in the beginning, but so Scott also sends hella specimens from Florida, North Carolina, Georgia, you name it. Scott goes around, travels in his car, sleeps in that son of a gun, wakes up on a trail. Yeah, Scott's a huge contributor as well as all the other things Scott does.
I heard he just lays down in a field and waits for him to grow up around him. Is that true?
That could be true.
I'm kidding.
But I do know that he parks on the trailhead and sleeps there. But so yeah, so high quality observations on iNaturalists. Maybe we can touch back on that. That's where it really starts. And then once you have the high quality observation, and I don't want to confuse people, high quality it just means good photos and like that you have an observation. That's very crucial. And then yeah, from there they need to follow certain packaging guidelines, which may seem a little strict, but I can explain why it is strict. And it's as simple as just mainly like a certain size bag, putting the certain information on the bag. I really don't care how it gets shipped. It just has to come here in a whole piece. I think people have all different unique styles to wrap and ship things, and that's okay. But it has to come in a certain Ziploc bag, and it has to be labeled with essentially one number, which is that unique identifier, which is the iNaturalist number or mushroom observer. But from there is when, if all those things are done, so the high quality observation, the specimen was dried the right way, cleared of debris, and put in a right size Ziploc bag, then that makes the person that is doing the tissue collection, which by the way is our largest bottleneck by a landslide. All the other stuff can be done relatively quick with a few hours of time, waiting on the bioinformatics, which takes upwards to 15 hours. But what takes the most time is the tissue collection. So we have to really stress on that. We did a really big open call 2023. You did see it. I mean, mounds, boxes everywhere. I mean, I literally was getting packages, multiple packages every single day, Monday through Saturday. The post office lady and I, we became good friends. She talked to me about mushrooms. Um, and that was great. And that was what really got us where we were with these numbers that we're presenting in the paper and on GenBank. But I can't say that it was the easiest. So, we learned from everything that we do. So we learned from that one. And what we basically did is we tailored the protocol a little more strict, a little bit more specific, to capture those very important things for the average person to be able to do to qualify to get their specimen sequenced for free. And so we're actually about to release that protocol. We'll probably do it when this podcast airs. And we will, send an open call for specimens again. And I do believe that we will get, more higher quality specimens for us to be able to utilize.
Love it. So if I send you a specimen and it doesn't have an INAT or a mushroom observer number on it, basura, right? That's
Yeah.
what are you gonna do with it? It's
Yeah.
just gonna be
Unfortunately, yeah, that is the protocol that we have in place. Is if it does show up and it does not have the unique identifier, that's a no go. That we can't work with that because I can't really, the whole trust system. Like one, I mainly probably won't know who it is. The only packages I really recognize are like my close friends, like Scott and Sarah and Jess, and maybe names I know. But like I said, we were getting mushrooms from everywhere, including Australia, including I don't even know, like Canada, Mexico. It was pretty crazy. So yeah, unfortunately, if it doesn't, we have to pitch it. I do not have the funds, the time, the resources to locate that and ship it back to someone. So unfortunately, it will just get trashed. Another case of why a specimen would get trashed is if it shows up completely contaminated, like fresh, right? Like if the mushroom, we've all seen it before, you don't get the mushroom dehydrated, you throw it in the Ziploc bag thinking it's good, check on it a day later, it's got all kinds of wild stuff growing on it. Just wouldn't make for a good process opening something like that in a lab space. So, and yeah, I just don't want to deal with dirty, mushy mushrooms. So that would be another case of the specimen just making it to the trash. And I don't like to do that. So we really, advise everyone to do the best to follow the protocol because throwing mushrooms away breaks my heart too. But it is obviously the only thing we can do when they show up like that.
right.
Can I chime in real quick with an important point?
Yeah.
Yeah.
That we like to remind people is if it's really an important specimen to you, to don't send the whole thing, because like Kyle said, you can't get it back to you. And so splitting that sample, saving some for yourself, and just sending out enough. And like Kyle only needs a little bit. It's truly a little tiny speck that we need to do this sequencing. Like people should definitely keep that in mind.
Yep,
Like,
for sure.
yeah, so for example, I got this Gymnopilus luteus that I found. The mushroom was about, I don't know, maybe that big. And
Good. I'm glad you're explaining this.
old Geeky would just, I wanted Kyle to just see my baby, the whole mushroom. So I would send him the whole, I mean, maybe if I found a horse agaric, I wouldn't send like the whole horse mushroom to him, but I usually send him the whole I sent him entire boletes. This is like radically way too much. Or sorry, I don't want to. Why can you not? Right, like
But
that's
very
way too
acceptable.
much.
Yeah, but very acceptable. That would
Right.
be, fine.
Yeah, you don't want it too small where Kyle's like, "Where is the mushroom?" But
Yeah. Oh, I got a couple of those too. Some little tiny mycenas, but yeah, you're just plucking the tiniest like tweezer bit in there, and that's gonna be more than enough. Right.
Yes, yeah, that specimen you just showed would be
Yes.
more than enough. But I
Now
would
I give enough. So I'm like, so with this luteus, I'm just like, I put the putative name on it if I think I know what it is. Sometimes I have no freaking clue. But this way, I'm giving you enough where I'm like, you can kind of get a sense. Oh yeah, it's like the right color. I can kind of see like maybe some gill spacing or something. And if the run doesn't go well, you definitely got some more to grab off of it. But yeah, you don't want the whole dang mushroom unless it's teeny, teeny, tiny.
right, unless it's super tiny or unless I specifically requested the whole mushroom, everybody sending for sequencing could assume that
Yeah.
a
You do that with psilocybes, right? You request the whole mushroom.
Yeah, that's my tip. That's why I just sent him the whole one as a tip. Yes.
yeah,
Here's a little extra for
yeah, I think I'm getting some tips. Yeah.
yes. Yeah.
Um, no, that's funny. But yeah, good bring up on that, Zach. Good call. And then Geeky, good on showing that sample. You are right. It is more than enough, and it's too much. But that size that you showed on a specimen, as large as a Gymnopilus luteus, is perfect.
Yeah.
Because some like Zach said, I don't want it to be too small. Obviously, with these super small mycenas, or any of the small stuff, what we say is, well, if that's all we had, then that was our
Yeah.
only attempt. I know that Sarah sent me super tiny stuff, and it's right in the tube, and we hope for the results to yield off of
Right.
that.
Yeah. And then I figure, right, like, I mean, I did on this one to figure out what it was, I did a little microscopy, but there's still some gill tissue in here. So if you guys get on the back end where you're like, God dang, I wish I could have looked at this under the scope, you can go back to a little bit of this tissue and maybe do some of that. Who knows?
Yeah, and that actually we do. So there are things that are in queue to be dug out and or already have been dug out. I know Jessica last time she was here at the lab, she took some stuff that we had previously sequenced, but because there was enough specimen, like tiny split like you showed, we got the sequence back, and it's novel, and now it's time to do micro, and we're able to resurface that specimen to do a little bit of microscopy. So it is helpful to have enough to still save, but not too much, obviously.
Right. Yeah, that's good. I think because, dude, it's not going to get less popular. It's going to get more popular. More and more people are going to want to do this more and more often. Man, I got some just straight up cube dudes who are, I'm telling you, they're wandering around the woods now. I'm hearing them say more genuses than, or more genera than just psilocybe now. So it's, not everybody gets the bug, but more people are getting the bug. Generally speaking, the interest in fungus overall is growing, especially fungus that has medicinal, gourmet, some sort of benefit to humanity. People go, I want to eat this one, or I want to ingest this one for psychedelic properties, for medicinal properties. The interest is not diminishing. This bubble ain't about to burst. This bubble is just starting to swell. And so, yeah, I really hope that I hope you have more and more work to do, more and more problems, because then that's going to have somebody go, shit, I've been watching Kyle complain about all the samples he's got for a year now. God dang it, I just got my tax return back. Maybe it got by a nanopore. Like I
Yep.
truly believe that there's gonna slowly and steadily be more and more people doing
Yeah.
this. I also think there's gonna be more people like Zach who are gonna come in from other angles and go, oh, this is cool. I mean, because E. coli is cool, but it's not that cool. Smells nice, but
yeah,
you know, it's
Yeah.
not sexy.
we're already on that progression. Like I think from when I started to see, I'm on all the same forms I was when I started, and now we're seeing a lot more individuals. Hey, I got the Nanopore. Hey, what's my next steps? Or hey, I've done this with the Nanopore. So yeah,
Yeah,
it's
weren't you telling me about some dude in Pennsylvania just got one? Or
I'm not sure,
no?
but
Oh,
I
maybe
will
not.
tell you that someone did just recently get them. Yeah.
Yeah,
You
we're
guys
all in
gotta
like
get you
okay.
got a private Nanopore group?
Well, I was gonna say we're all in like the same like Facebook groups, and every once in a while you'll see someone post like, either they just got one or they're like, hey, what does it take to get into this? And you don't have to go full hog into Nanopore. There's like kind of beginner setups if you wanted to like ease your way into just like learning PCR at home. Because basically everything you do for like Sanger is transferable to Nanopore. You could, find something that you're really interested in. It does cost, a good bit of change, especially going to like something at the Nanopore level. But for some people, like it's worth it. And if you're really that interested, like, definitely something to encourage like the more people who could do this. And again, like everything we do are data pipelines and stuff like that are openly accessible. Like we're not holding any sequence. Actually, that paper I mentioned of our kind of like the first half of uh, what we have done was also like the paper was presented as kind of like a blueprint of like this is how we did it and how you could like replicate this if you wanted to.
This is we talk about this in the cube community too, which at least this is a thing I believe, right? It's a burgeoning community. It's not wide scale. Everybody on Earth doesn't know what cubensis means, right? Like I can't walk into the grocery store in Ohio here and pull 10 random people and go, "Do you know what cubensis means?" Most people still don't know what that is. Or, I mean, and I can spot the ones who might have a clue pretty fast. But we're not far away from that being a more mainstream word. Psilocybe, psilocybin, these are slowly growing to be more mainstream words. And so I always talk about how talk to people about this because you might talk to 20 people in the next year about this, and 3 of them might be just waiting to know about this. And so the
Mhm.
same with Nanoport. The more Zachs we got, the more Kyles we got, the more Steven Russells we got, the more Alan Rockefellers we got, the more people we got that give a shit about this and want to do work around this, the quicker y'all figure stuff out. You figure out new protocols. You go,
Mhm.
oh, we just got this new lyophilized primer, and let me show you a trick I figured out with it, like that, right? Lab secrets, Zach. You know all about this, right? All these, like, there's the stuff that gets published in the paper for transparency reasons, but then there's also like the little slick things that they definitely don't want to tell anybody they figured out how to do.
And even worse if that paper is locked behind a $50 paywall, like props to Stephen Russell for putting out like on protocols IO, a popular like open platform for protocols. In like I've been working more with Steven and helping him iterate on like version 4 or whatever of the data pipeline and also the molecular methods. Like I've been talking a lot about the software side, but I also come from like a background of designing PCR and qPCR methods and stuff, and I get really geeked about that because there's again just like continuous improvements. Like there's always something to be working on.
Because you can't do it all by yourself. I mean, you can, and every once in a while, if you do, you get a Nobel Prize. But generally speaking, many hands make light work. It's it makes it faster. I mean, you gotta have more people doing all this stuff. So, all right, let's do this. Let's tell the story because I've had people who, because we talked about Michael Blitz from last year, they submitted 10 or 20 samples or whatever. And then I get somebody message me and go, yeah, it's been a month and I haven't gotten my sequences back yet. And then I got to tell them, yeah, me neither. I've submitted, a couple hundred, I think, and I don't have them all back either. Maybe I have 2 or maybe I have none, or now I got 40. But instead of just saying, be patient, I was talking to Kyle. I said, let's just really paint a picture of from the time you stumble across a mushroom in the woods to Scott or Alan or somebody or Jessica or Sarah or now Chris or now who did you just add who's doing validation? Sigrid. Yeah, sorry. That's right. Right. Somebody. Oh yeah. I'm like, oh cool. I'm glad you got her. She's cranking through a couple of my things. This is great. Um, like just to see what goes into it and to see how like yeah, there's stuff I've sent Kyle that I might not get a sequence back on. I know why, but some people don't. So let's paint that picture. Step one, somebody like me or Scott camping out on a trailhead waiting for the mushrooms to grow around him. Kyle wandering around the woods in the dead of winter thinking he's going or I don't know, tree identification, not even looking for mushrooms. So we find a mushroom. So here's my mushroom. What should I do? What is step one for me? We will assume that I have iNaturalist, but what should I do with this mushroom if I think I want to send it to you?
So ideally, you I like to take pictures in situ or in habitat. Like before I remove it, I try and get good photos of it in habitat. And then I don't do this as much as I should, but like a farther back photo of the actual habitat, like so it's in context, is definitely useful. And I love seeing that. Um, you know, I do sequence validation for fundus, and they have like pretty high standards for their iNaturalist observations for their collectors. So they also do like a KOH reaction on their mushrooms.
On
They do,
everything.
yeah,
Yeah.
which is great. And if you want to go above and beyond like that, I would encourage that. But obviously, not everyone is there or cares enough. So, yeah, UV reaction is great. KOH reaction is great. Scale bars, that's extremely useful. Especially all these additional things are extremely useful if you want to make your observation useful to mycology in the future, especially if you do come across something that is novel and can potentially be described, having those data points is just a great addition to have. But so, yeah, you'd take a picture in habitat before you remove it, and then you'd want to remove the mushroom. And depending on the substrate and like the species, I guess Amanita, for example, they tend to have these vulvas that are like under the soil. And so taking a knife and extracting it from the soil, ideally unbroken, so you can get a photo of the mushroom in its entirety. That's extremely useful, especially for that genus where like, you can get it idea to section just by looking at like the annulus and the vulva and uh, trees it's growing with. Anyway,
Well, and so I should also say, when we found Neohalopensis in Mexico, the coolest thing was realizing how deep that stipe was embedded in leaf litter and all that. So there are for sure the amanitas, but sometimes just seeing how deep that stipe goes into the soil or whatever substrate it's growing from can also be useful.
and then getting a shot of the gills. So something I dislike seeing is like partially dried photos like sitting on top of a tackle box and the photos taken from like a distance, because when I get to it in the validation process and I'm trying to match up the sequence to it, sometimes it's hard for me to tell if the sequence even goes to that record. And this is actually like a little bit of an issue because Nanopore produces something called crosstalk, where you can pretty often actually have multiple sequences attached to a single record. And for me, that's like a black box of I don't know how that happens, and I know Zach and Kyle and Steven have more insight into that. But sometimes that is an issue, and sometimes that's the reason you're not getting a result back is because it's sitting in the spreadsheet as come back to this one because I can't figure it out, and usually I'll have to consult. Like if it's an Ohio species, I'll consult Jessica and say, hey, like, do you think this sequence for, Gymnopus, whatever, Dryophilus goes to this observation? And usually she's helpful and can confirm that. Yeah, if it's a taxon I'm not familiar with, sometimes it's hard. So anyway, just painting the picture of the importance of like good quality photos and, if you can include as many fruit bodies at different stages in development in your photo as well, that's extremely useful. I guess not as relevant to our work, but something I like doing is having little tags, like numbered tags that I set with the collection and take a picture of. So before it even enters INAT, it has a unique number that I can associate it with. And then when I put it in the tackle box, I will insert the slip into there as well. So that if I'm having a long day of collecting and I, make many collections, it's all organized and I can refer to the photos with the numbers on it. And I know which collection is which, especially if I'm putting it on my dashboard in between trails and it starts drying out and dehydrating. Sometimes it's hard to discern which. Yeah,
you can forget what that thing is. Yeah, some of them you're like, how did it turn into that? Like what? Yes.
And then on top of that, and I actually need to get me one of these, but ventilated tackle boxes. Because sometimes if you have a tackle box full of mushrooms, things like, especially amanita and boletes, if you close that tackle box and then set it in your car for a couple hours, sometimes it'll just turn to mush and especially if your car's not on, having that ventilated tackle box will prevent that happening to a degree.
Oh yeah. Some will also just blast out some spores out of nowhere if they sit in the box too long too.
Yeah.
I didn't even know there was a ventilated tackle box. Now I'm like, oh.
I didn't until last week. I met up with someone and this guy had a vent. I was like, oh my gosh, I need I need that in my life.
Nice. Nice. More things I need to buy.
So, really high quality photos in habitat, as many specimens that you confirm are the same species in the same photo. Those are all great. And, for the people who have been sending Kyle stuff and you know who you are, I would encourage you guys to consider integrating some of these other metrics like KOH reaction, size with a scale bar, and UV reaction. If you really want to go full Alan Rockefeller, get into focus stacking. Yeah.
Man, I can tell you with my gill porn game, I used to just think I'm just gonna get like this, nice little upskirt, get most of the stalk in there. And then I finally realized, well, what I really want to see, what you guys really want to see is you want to see how the gill attaches to the stipe. And you'd like to, if it's possible, both see the density at which the gills are spaced. So you don't actually need, depending upon the size of the mushroom, you don't have to have the entire circle of gills present in the photo. You just have to see at least somewhere the stipe and somewhere an amount of the margin, unless there's something unique somewhere else going on. And getting closer helps you see sometimes spore color, little other, margin information, stuff like that. So one thing, and it was the minute you said this, it popped in my head. I used to be the guy who was lazy because I found a mushroom and I no more than find a mushroom and I'm trying to collect it. Then I just look over like that. And now I see another mushroom. So I get just, the dopamine's just hitting and I'm just going and my ADHD is just having a blast. But take the fucking time and get all your photos. Don't rush it, because
The mushroom will still be there.
yeah, it's right there. It might even be cuter by the time you wait just another minute. You never know. Yeah, is get all the good photos and get really close. Don't be afraid to get some really close up photos. Some boletes, you want to see the reticulation. Some things drop spores in a particular way where if you take photos of the way spores have fallen onto things, just all the little unique photos. You can't take too many photos.
Right. And something that can really inform how you take photos is read a species description.
Yeah.
If you do that, like, I think people might underestimate like how much detail goes into that, and like, because everything, I mean, at least like good species descriptions describe everything, and you will walk away from reading that a lot more informed about what you should be paying attention to when it comes to photographing your mushroom.
Where do you go for that? Like MycoCo's
Mushroomexpert.com is probably not the worst starting point for most people, right?
Yeah. I mean, or a paper describing a mushroom.
Yeah.
Oh yeah.
If you really want to be intimidated, yes, you go to like novel species paper that is extremely detailed. Yeah. But I think you're right. And then one thing that starts happening slowly, right in the beginning, you learn a couple things and you go, holy shit, how am I going to learn all this? And then pretty soon you start, for different clusters of mushroom types, you have different strategies for how you photograph it. And I'm even getting better at trying to figure out what is for this particular type of mushroom morphology, what is like the magic shot for iNat AI to give a better guess at what this is. Because man, you can, and the top view of the mushroom 90% of the time is not that shot. It's not that shot. But then for polypores, sometimes that is the shot. But until you start playing around with this, sending stuff to Kyle and finding out, what guesses are good and what guesses are not good, you don't know. Take a lot of photos. You can never go bad with that. If you upload 20 photos for one observation, and the one you use to try to identify that mushroom didn't get you there, then you can go, okay, cool, Scott just told me what it is, and then you can go play a game. You can take that other photo and try to make new observations and see if any of those other photos properly identify. It's a game I've been starting to play just to try to figure out, because the AI sucks right now. It's not, I mean, it's good, but it's not. In 5 years, the AI for that's probably going to be bonkers. It's going to get way better, and as more people use it, it's going to get better. But yeah, that's
maybe in 5 years the AI could identify an anosogeny, but right now, no.
Right now we need Kyle. Yes. Until then, yes.
I want to share a prop real quick. So something I started doing, because I have a large collection of tackle boxes that I want to hand out to people, so I started drawing scale bars on them.
Oh.
So that way I don't have to take like a ruler or something. I can just like take the in habitat photos and then I can place it on top of the tackle box with the scale bar. And this one's done in centimeters. Then I have that. So one less thing in my pocket or bag.
So we can't use just a Bic lighter. That doesn't count.
You can.
Better than nothing, I guess.
That's what we do in the old cube community. It's just always put a little Bic lighter in there. Just
oh no, I mean, I definitely see collectors doing that. I see people using coins, buttons, whatever you've got. But this is my new favorite.
I like
a
that.
3D printed toad like Sarah has, a 3D printed toad. That's pretty cool.
Oh yeah. Yeah, I
Yeah.
like that. I'm thinking, I wonder, every once in a while, these sticker companies do translucent or like transparent stickers. Be cool to just get one of those scales made and you could just send out slaps. People could slap them on their tackle box because that
Yeah.
level of commitment to draw that out is probably exceeds most people's capacity, unfortunately.
If you were really dedicated, you could be like Joey and get the tattoo on your finger or your hand.
Oh, right. Yes. That is
Doesn't he
hilarious.
have that? Doesn't Joey have like one of the scale bars on his hands? I'm pretty sure he does.
I think
Yeah,
so.
that
Yeah.
is, I don't know super how accurate that is. Maybe as he gets older, it's gonna get less accurate, but
Yeah,
cool.
probably.
It's cool nonetheless. It's definitely cool. All right, so let's assume we've taken a bunch of great photos, and let's also assume that unlike a lot of people who watch the show, they don't have a dehydrator at home. So how should one dehydrate mushrooms of various sizes?
That's a good question. And sometimes
I can.
it's hard for me to relate to that. I have like 4 dehydrators. But if you have the tackle box and you're, out all day in between trails, what I always do is I'll just like open up the tackle. Oh shit. I just broke the clip. Anyway. I just have it open on my dashboard and the sun, at least here in Florida during the summer, it takes care of it pretty nicely. And a lot of the times I'll have at least like 80% dehydrated mushrooms by the time I get home.
Right.
So you can at least partially dehydrate it on your dashboard. Um, Something I used to do years ago when I used to collect cubes is I would have it sitting on top of something I would get it as close to my fan as
Mm.
possible on top of some like paper towels so that it was getting like wind or circulation regularly.
Sure.
Um, yeah, get
I
a
have.
So
dehydrator.
I guess I'm getting at like, and do you think people can dehydrate these without a dehydrator sufficiently enough so that you have a quality sample?
Yeah. So one technique I used to use before I also had a dehydrator was an oven tech method that I picked up off some YouTube channel that's now banned and gone. So that's unfortunate. But I found 100 degrees, and you set them on a cookie sheet or aluminum foil, whatever, and crack the door open. Obviously, just be very cautious, but it's not going to hurt nothing. And that was actually how I dehydrated everything that I successfully got results for off my herbarium. Off my herbarium. So I knew how it
Yeah,
worked.
I've done that too, and like sometimes the oven doesn't go down to like
Like as low as 100, right?
oh yeah, like when I was at my parents, it went down to like 170 or something, and so like Kyle said, I just like left the door cracked a little bit, and it worked really well. Also, like I have an air fryer, and I know that has like a dehydrator setting that'll go down to like 140. Because if you do have a dehydrator, I think like what 130, 140 is like a good spot to dry it out. Like it'll go pretty quickly at that temperature.
Yeah, I cook them at 165 and they
Same. Same. And I'm glad that you brought that up because I cook mine and my sequences come back. But if I was telling the average person, they might think it's biased. But your sequence also come back, and they're cooked. We have people from all over, and we have people like in Florida and California and places where it's hot. And I know for sure some of Scott's stuff probably was never dehydrated. It was just dried, naturally on his dashboard. And I know the other folks are the way. So I think it just depends on where you live. But ultimately, just get a dehydrator. They're like $20.
I
Um,
mean, literally on Amazon, I think you can get a pretty solid one for like $30 shipped to
yeah.
I
your house.
picked one of mine I got from Goodwill for $5.
Ooh,
There you
see right
go.
there.
Yeah.
Yeah. Those kinds of
Man,
stores.
I got a $350 one off Facebook Marketplace for $65. So
Damn.
also Facebook Marketplace, yard sales, Goodwill, get creative. But at the end of the day, $30-40 gets you a pretty solid unit off of Amazon next day delivery, guys. So yeah, if you're doing all this work, it's not that much money to dehydrate, and then you can make beef jerky. You can dry out some mango slices. Come on, you can get your money's worth for sure. It's not a problem. So yeah, so dry your samples. You guys can't have them deliquescing or, man, I had a horse mushroom that I had sitting in it was like this freaking big, sitting in my garage for one afternoon. I went to go run an errand and I looked at it when I left and it was fine. When I came back, it was like, blah, just like that went off. So you guys get a sample. It's properly dried. It's got an INAT observation number on it. It's dated. It's in the right Ziploc bag.
So, as I mentioned before, Scott's collections were some of the best that I had in the beginning. And I don't know, it just might have been a coincidence. And so we fell in love with his packaging, and I know there's other folks on the team. I know Zach's done sampling, Jessica has done sampling, Scott's even done some. So everybody has gotten a taste of it, and I think most of us agree that there is actually a science behind it that helps doing the collection. Like you could take a whole plate, 45 minutes with good collections. Say there are a bunch of random collections and you're trying to build a plate of 96, it might take you an hour and a half, and it doesn't seem like that big of a difference. But again, we're talking high throughput, so it's not one plate we're building. So yeah, he's going to show a good example of the Ziploc bag we use. And I know Scott also put some extra information on there, and that's helpful. But again, just for the average person wanting to learn, we only need the iNaturalist number on the bag. That's it.
Right. So, I I think Kyle brought it up before, or maybe you did, Geeky, but splitting your collection and not sending the whole thing. So I have the master bag, and shoot, this glares.
Yeah, I could see.
But yeah, so I have the INAT number, I have the genus it belongs to, I have the date, the state, and then I have other finger, my unique tag number, which is number 316. And then once I split it into the bag that I'm going to send to Kyle on the master bag, I'll write with Kyle. Let me see. I write you get it. So that way I can keep track of what I've sent to him. And if I go back into my fungarium, I can see if I missed one. And then just so this would be like a bag I sent to Kyle. And in this case, it was a really small mushroom growing on leaf litter. So this is the only bag I have of it. And I'm just sending the whole thing. But, INAT number, right now it's at Agaricales, date, Florida, my number. And, this other information is helpful, at least for me, because I've occasionally forgotten to write the INAT number, and then I've just written the rest of the info. So, I don't know if Kyle would do this for everyone, and it'd probably be impossible to do because it's hard to track what bag was from whom, but Kyle knows my bags and where it came from. So if that does happen, he can take a picture and I can see, Agaricales from May 16th of this year, and my code is 391. I can quickly sift through my INAT observations from that day, find 391, and be like, hey, this is the INAT number.
That's the point of the date. I like that. See, I don't know. I'm OCD. I'm gonna get the INAT number. I go in order, so I have maybe 40 wells in one, and I always load them the same way. I have the hinge at the top, and so I have my own little process, but I think it's important to say you gotta have a process. If you don't think through all this stuff, you will get home, and like Scott said, some of these things will be dry and look different even just by the next morning, and you'll go, wait, is that? And the white and the brown ones, the medium-sized white and brown ones, all start looking the same when they start drying out, and you're like, is that one this and this? And if you don't have a process for all that, yeah, you can screw it up. And do not send a mushroom that you're not certain you got the right INAT observation for, or then you really make Scott's job difficult.
And then legibility as well,
Yeah.
because it's actually pretty common that we are in the spreadsheet and we pull up the record and it's like a fish because one number is off. And occasionally I can figure it out where I look at the rest of the plate and I see, oh, it's from Ohio and it was all from the same collector, and the sequence comes back as Mycena. I can go to that collector on and this is when I'm really feeling like going above and beyond, but I'll go to their INAT, I'll sort by their Mycenas, and yeah, I'll see which INAT is one digit different from the one we have that turned up as a fish or a plant.
You just made me feel so good about my OCD, dude. You just made me feel so good. That is another level that I don't know, but you want to figure it out, right? This is like, I was talking to Kyle about this. It's like why people like murder mysteries, right? They want to find out who did it. You want to find out what it is. You're like, oh my God, we did all this work. Am I going to get foiled by a stupid, typo or how somebody writes their frickin' sevens doesn't look right? No, I'm going to figure it out. So date, state.
I mean, that's what I do. I don't think Kyle expects everyone to do that. The most important thing is an INAT number and legibility. I think I just fall in that OCD category, and I like having that information.
Even you saying like, have a ruler or have some way of rough measurement. These are just little things you just got to incorporate into your bag and into your process, or bring a little tiny bottle of KOH out is no big deal. And sometimes it is clutch for figuring stuff out fast. So why not? Why not? If you're going to do all this, if you get into it, if you like it, if you're not sending like just your 10 Myco Blitz submissions, but you're sending 50, 100, if you start getting into this, that's when you got to start thinking about making life easy for Kyle, making life easy for Scott, for Sarah, Jessica, Zach, everybody. Like, do just a little bit more work on your very front end to help everything go more smoothly for these guys. I mean, it's just courtesy in my opinion, but you don't have to do it. You can be an asshole. You can just send them a bunch of stuff with shitty handwriting and but then when you don't get your sequences back, don't be surprised.
And then another thing that me and Kyle have been discussing recently, as well as I've had a conversation with Steven about this recently as well. Like if you can establish in the state that you're in, assuming you have like kind of a community of collectors, like obviously Ohio has, establishing like a triage person who you can send everyone just sends their stuff to Jessica in Ohio, and then she can kind of do like a filtering, like quality check of everything, and then send it all at once in one package so that Kyle doesn't receive.
Yeah, we're using Sarah actually. Sarah's going to be the triage for Ohio.
Okay. Cool.
But yeah, I've been doing that recently for a couple of people at University of Florida. I actually just got like 100 specimens from the Everglades. So, and I don't think anything's been sequenced from there. So I'm excited.
Let's go.
I like that. Very cool, man. All right. So we take good photos. We get the right numbers on the right Ziploc bags. We send everything to you guys. If there's a triage person in the state, we send it to them. So now, Kyle, let's assume you have a good sample. What is your timeline, your workflow look like?
All right, so this is the question everybody wants to know. So I got my phone out. I was doing a little bit of math here. So upon receiving the specimens, now that we have a triage, that kind of takes out a little part that I had to play doing that. So let's assume I get a box from Sarah and it has 500 samples that had been sent to her over like a month period. Because we are, the whole reason we are doing nanopore sequencing, I mean, there's multiple reasons, but the big reason is because of the output that we can achieve. So, but with that, doesn't necessarily mean when you hear high throughput, it doesn't necessarily mean like we get the results very quick either. Can you? Yes. But we're going to explain why our lab does not do these quick results. And the main reason is cost. And so we are a free sequencing lab. We will remain a free sequencing lab. So we have to have everything in order that makes the most sense. And with a box of 500 mushrooms, that's actually not even enough to load a fungal flow cell. Can you? Yes. And have we? Yes. But now that we are really ramping up, that is not the plan anymore because we need to maximize the capacity of the chemistry and the fungal and the person's labor. So they will go into a queue, and the queue is basically mushrooms in plates or in the bags. They get plated out by a specialist, is what I'm going to call it, because it's not an easy job. And those people are like myself, Jessica, and most recently Brian Hunt in Indiana. So Brian Hunt's a guy that has been doing this for Stephen, and Stephen graciously donated some funds for Brian to do it for our lab as well. So that's going to help. So once we receive enough specimens for a run, which right now we are running 8 plates, and that is just due to let's just say Stephen's robot right now, because he's indexing the primer plates that we use, which are those tagged primers that you and Zach were talking about earlier with polymerase, and then that's what we use for our PCR reactions. So right now we're at 8 plates, which is 768 mushrooms, I believe, 96 times 8. So I will need at least 768 mushrooms to run in one flongle. So you could kind of see it takes a little bit of time. When I've been on Facebook telling folks to be patient, this is kind of what I mean. And now Nanopore has changed whether they changed it or they're just now following it, a new warranty policy for the fungal flow cells is 4 weeks. And yeah, that's what I mean, I can't say. I don't know. There's a lot of like different folks saying different things, but I'll be honest and say that nobody knew it was 4 weeks. The 4-week thing kind of shook the community, but so now we're limited to this warranty, and you can't buy just one flow cell. So you have to buy the flow cells in packs, and the pack is 12, and they can do 2 increments, shipping. And so you can get 6 flow cells, and then a couple weeks later you can get 6 more. So you take the 768 and you times it by 6, and now you're looking at 4,608 mushrooms. And that's just at the 8 plates. Mind you, Stephen Russell has ran 10 plates on one fungal before, and there is talks of us gearing back up to 10, maybe even 12. So I would roughly need 4,608 mushrooms to make this successful now with this newer warranty period. And 2023 was our year to try everything out, and we did try, and we did come up with some success, but we also had a lot of failures and a lot of things that we need to readjust. So this is round 2. Here we go. We're going to start sequencing here this month, I believe. Don't mark me too with it, but I'm thinking, yes, by the end of the month, we'll be sequencing.
Oh, it's happening, Kyle.
Oh, it's happening. I mean, we get a large number of plates built up, and that's what we're doing right now with Brian, Jessica, and myself. Is I have officially gotten rid of everything here at the lab, and it has all been sent to Brian and shipped to Brian's house. minus a few things that I'm going to do the tissue on, build about five more plates myself. And so, yeah, we'll be looking to doing the sequencing in June. So again, sometimes it just works this way. Like I know there were folks that had sent stuff, and I know some of the people sending stuff, and then there's some people I maybe never had a conversation with, but they follow us or me or whatever, and they ship stuff. Um, and so in the beginning, there was like whoever shipped stuff in the very beginning, they likely waited longer for results. And it was due to waiting until we had, a large number to start processing. And then I know that there are folks that just got very lucky and they were one of the last people that shipped their packages in, and they got results in like 2 weeks because it happened to be at the end of the tissue collection, and we had enough samples to put through the fungals, and 2 weeks later there was results. So
One of the cases where the slackers got rewarded.
yeah,
Timing was
or just
right.
luck of the draw, really.
Yeah.
So there is a time process on it, and I think we never did guarantee anybody results in a timely manner. We just basically said, hey, we will get you results, and I'll talk about a little bit about why you won't get results, and then we can let Zach wrap up the Nanopore stuff. So if you waited and you did not get results, I have a little checklist in the protocol. So once that gets re-released to the public, you could follow the checklist and just give you a better understanding of why. But as we had already mentioned, if it showed up moldy, rotten, full of water, no iNaturalist number, that would qualify for the specimen to not be sequenced. And then, so that's one. And then two, your sample has been collected and we did attempt to sequence it. However, it was within that percentage of failures. So to make it very clear, when we do these runs, yes, we are trying to do 768 samples, 960 if we're doing 10 plates, but we do not expect 960 sequences. We don't even expect 900. I don't even think we expect 800. So we do know that there's going to be failures, and so unfortunately, your specimen could be part of that failure. And there's later plans, and I don't want to say because I don't know yet, but we do want to try to rework some of these failures. And then what would be another reason it failed or you didn't get it? Yeah, the what we call fat fingers or typo errors. Those also play a role. And like Scott mentioned, we do go above and beyond because at the end of the day, this is what we want to do and we do care. So yeah, so we're going to try to go the extreme to get the sequence, but obviously there's times where it doesn't matter, or it matters, but we've exhausted all options and we can't come to a conclusion. So it gets put in the queue, gets sequenced, and then there is a backlog sometimes on the bioinformatics. Sometimes we're ahead or we have certain things we want to look at in a run. So it could get sequenced and then it could take, a little bit of time for the bioinformatics to come back. And then the validation, right? So that's another bottleneck and limiting factor. We have great people doing it. I mean, we really do. We got Scott and Sigrid staying up. I don't even know if they go to sleep sometimes. Jessica, other great validators. So it is a little bit of a process on every single end. What I tried to guarantee everybody that sent me sequences or sent me specimens in 2023, I said I'll try to get sequences within the year. That didn't work out, but I do believe by the end of this year, everything should at least we will at least have attempted a first pass, and yeah, there's maybe even talks about like releasing a little document to the public too, because we work with some of our like Google Docs spreadsheets that we work with that do the analysis. But there's one that we have that I can consider sharing to the public that it wouldn't impact us if something happened to the sheet. Obviously, I don't want nothing to happen to the sheet, but we can protect it. And essentially, you could match your number to the sheet and kind of get an understanding. So, that's what I do when people will say, hey, what's the status on this? I say, give me the number, and I run the number. And if the number is in my sample sheet, then I tell them, hey, it's been sampled, and continue to wait patiently and hope that the results come back. And then there are cases where folks will send me the number, and I will check the data sheet, and I will say it has not been sampled. Now, once Brian Hunt completes everything from 2023, which will be this month, I will send a little blurb out to everybody, and they will have access to check. And then if your number is not on the list, then we're going to assume that it didn't have the number, it was molded, whatever the reason may be, it did not make the cut.
I mean, I look at it like this, right? It's like, you ever see at work, right? They have free pizza and nobody ever goes, well, what's on it? What's this? What's that? I don't like this. I don't like that. It's free pizza. Now, if you're ordering your pizza at Antonio's or wherever, then you're like, I don't want this on it, and I only like honey if it comes from San Diego, California, and I don't want this on, right? But free, you don't get to complain as much. You guys are making every attempt with a quality sample to run Nanopore on it. A lot of times it works out, sometimes it doesn't work out, and that's the game. If you're doing a very significant bit of scientific research with your sample and you're sending it off to Kyle and you're getting mad because you didn't get a read back, hey, you can also have it run Sanger. You could probably pay Kyle to run it Sanger. You can pay a bunch of different people to run it Sanger, and if it really freaking matters, you can pay them, I don't know what, $5,000-10,000. You can get Illumina run on it, and
Yeah.
but it's free, guys. It's free. So you're. This is why I would encourage people: don't send 5 things to them.
Yeah, don't.
Send more things.
Right.
More
Yep.
things.
Because
You'll get.
the likelihood of you getting a sequence, sending more specimens is big. And that's kind of my goal for everybody, is to get them some sort of results. And yeah, and so I've also encouraged them, like, hey, send me more, because these could just fall into that category and not make it. But like, and Scott knows, and Zach knows, and everyone that's sent more than 50, I'm talking hundreds, or whatever, you guys know, you're seeing the results come back. Now, does Scott go to his eye naturalist every now and then when we're on a late night Zoom call and go, damn it, that never came back? Of course. But what do? We just keep going. There's enough good stuff that is coming back. And that's just the nature of the game, right? But nobody said that Nanopore was so perfect that you're going to get it all. Quite frankly, my first couple runs were like less than 50%. So, but
that's still so many reads.
we, like, my first run, it did tank, but we got so much data out of the first run, and it's what made it worth it. And when you understand that all these aspects of sequencing, then you really are like, dude, Nanopore is the shit. It's $1,000. I did it at my house. Like, I turned 2 rooms into a lab here. Like, I'm not in an academic setting. I'm just a dude, man, just trying to get results on something I'm passionate about. So
Another tip, staggering specimens. Let's say you have 200, send 100, 4-6 weeks later, send another 100. That way you can have like steady results, and that dopamine just stays going off. That's what I like to do. I don't like to send too much at once, because then I'm waiting. So I'll do like 100, 150 at a time, wait a month or 2. Yeah.
Yes, you're like piecemealing out your dopamine hits.
Yeah.
Yes. You're like, do you want to just log on to iNet one day and have like 400, updates, or do you want to just get 5 or 10 like every couple days? That's what I'm really looking.
Right, and another benefit of that is you can dig in to the updated records without like forgetting about them. If you get like 10 back one day, you can kind of like really dig into that. But if you get like 50 things validated and they're all cool, you won't know where to start, and then you're likely to just forget about some of them. And
right.
Yeah, for me, it's more send you just a bunch of stuff, and then when it comes back, I just get to, be excited about it.
Yeah.
I'm not I don't have too many where I'm just going, when is he gonna get that back? I do have a couple, and I'm still waiting. But I mean, I also, it's like what I teach my children. Bugging me is not gonna get you what you want any faster. So yeah, I know you guys know what you're doing, and you're working the process. So it is what it is. Man, when you were telling me that you had to buy 6 packs of your flow cells, I was like, Nanopore, what are you doing to them? Come on,
Mm.
man. Sure, just charge a little bit more, but sell them one for the love of God. Like,
Yeah,
you know what I
that
mean?
was
I get. I mean, they must just need to move product. It's got to be an expiration date thing or something. I don't know.
Yeah, I do think it's a money thing for or like money maker for them in some
Yeah.
ways compared to the other like full size ones.
I mean,
There's
compared to all the other sequencing technology, it can't be like king of the hill as far as revenue goes. So yeah, they got to be
exactly,
looking.
yeah, they got to make their money back somewhere. Like that it's the $1,000 to buy into it is heavily subsidized on their end. Like yeah, and like I was saying, there's like many reasons your samples could fail. There's so many moving parts through this lengthy process that something goes wrong somewhere along the way, and then that sample just disappears out of the end product. There's also, we didn't really mention it, but like certain taxa also just don't do well for ITS, whether that's like you get an ITS, but the species sequences are too similar to actually like discern, or some species just don't amplify well, and that's more of like a futile effort even from the start. And we don't know until we try, but yeah, like this process, not only is it free, and or maybe because it's free, um the protocols and choices of methods we're using are definitely optimized for efficiency and cost effectiveness and these kind of things. So yeah, like we're accepting that it's not going to be 100% effective. That's kind of just a given that we have to accept. But yeah, we're striving to make it possible, feasible in the first place.
Now, have you looked into? Because I know polypores don't always extract, or I don't know if it's they're not extracting well or amplifying well, or they're not nanoporing well. And what chanterelles also don't, right? But some of these like troublesome genera, like, do you guys have any idea why they're troublesome?
Those are two good examples because of two different reasons. Like polypores, especially like really hard ones, can be, it's an extraction problem. Um,
You just
and
can't get it out
you
of
can't
it
get the DNA out in a good way to make
without
it
ruining it or something,
for
right?
yeah, or it's just kind of it's just it's too tough really, like at a cellular level. But then your second example of Cantarellus or chanterelles, that's an example, and Scott would know more about this actually, of where ITS just isn't the best choice of a gene to be able to figure it out.
Right. Okay.
Yep.
Interesting.
So
Cool.
going
So,
back to the samples in the lab, is that where we're still at?
yep. So you're building up a run. You're going to do a run, and I mean, you sort of touched on sometimes that goes well, sometimes it doesn't go well for different reasons. But yeah, I think that's right about where we're at.
I'll go up to tissue extraction and PCR, and then if Zach, you want to take over the rest of the nanopore stuff, you obviously do it and have done it. So yeah, the specimens are coming in. Say I am the tissue person for 2024, which I don't believe. So upon the triage, it's going to go to Sarah. From there, it's probably going to go to Brian or someone else that is specializing in doing this. And they're going to be the person that's responsible for taking the tissue out of the bag, putting it in the corresponding tube, recording that number in a data set. That basically, when you see a 96 well plate, you're never going to know what's in there. But you have a corresponding data sheet that says, hey, in each one of these wells is this specific number. So it goes back to the whole thing of why everything has to be done the right way at the beginning, because now we're down to an iNaturalist number and a small piece of tissue that you're not going to be able to distinguish whatsoever. So you have to do DNA extraction without getting into too much detail. You're adding some solution, you're maybe macerating, and you're putting it on a heat source for X amount of time. So up to an hour is what I do. So again, I'm limited with equipment here at the lab. So, we're talking 8 plates for one fungal run. I have one thermocycler that'll do 1 plate at a time. And so with the extraction, each plate has to be extracted for 1 hour. And mind you, there's a lot of bench work, lab work. So now you're looking over an hour a plate. Once you successfully get DNA extracted, you'll transfer that into the reagents you need to do PCR, polymers chain reaction. And so that's where I'm definitely limited to one thermocycler. So I would have to cycle plate. And what happens is I get these little weeks where I know I'm sequencing, and I pretty much stay up late or set alarms. And every 3 hours, I'm waking up and pulling a plate out, putting a new one in, because it roughly takes like 3 hours for a PCR. And then everything gets PCR'd. And we still, at this point, we have no clue. We don't know anything. All we know is we have this tissue and this tube, and this number says so. And we attempted to extract, and we attempted PCR. At no point do we validate those samples, mainly because of cost. There is a very great validation process called gel electrophoresis, which, if you get into Sanger sequencing or you know much about it, which I know some of your audience does, then they know. However, running gel electrophoresis on 700 some samples that you attempted is going to add up really quick, and then it defeats the whole purpose of what we're trying to accomplish with obtaining a sequence for as low cost as possible, obviously still high quality. So after PCR, we still have no idea. It's just that attempt to amplify that ITS region for us to be able to have that DNA to go on into the sequencer. So what it looks like right before I'm about to sequence is I have multiple freezers and refrigerators in the lab, and one of the freezers will be full of PCR plates that have been done in the thermocycler. And now it's time to basically pull those out of the freezer and start this nice little awesome, very fun process that we call DNA sequencing.
And I was thinking, I already kind of briefly explained a little bit of that nanopore process, but just to recap, so like the idea of PCR is to amplify just that snippet of the gene we need exponentially, which is a key aspect of it too. So you end up with billions of copies of that same gene, and then you go into what's called library prep. And you do a couple other steps, like it's end repair. So you have just that snippet of a gene. You want to make sure the ends of it are ready to accept like the nanopore specific adapter sequence. So it has that affinity to the pores we were talking about to be sequenced. You're basically making the DNA compatible for the sequencer. And you're doing, a little bit of like QC check, quantifying how much you have. Like we mentioned before, it's just absolutely minuscule amounts. We're talking nanograms of DNA, which is, like billionths of a gram of DNA, and so you have to use very precise, it's actually a fluorometer. It's using fluorescence to quantify like how much DNA you have. So you're loading this very precise amount that's ready to go onto the fungal.
Because you don't, in this process of just extracting and amplifying to the nth degree, you don't want to clog your pipes.
Yeah.
You don't want to clog your pores. Okay.
Yeah, yes. that would be right. One of the worst things you could do at this point, especially after all the work you've done.
So exponentially amplify, but don't exponentially amplify too much.
Well, actually, so there's key dilutions in place. So you are amplifying, like you're definitely trying to get as much as possible from the start, but you also know you're going to dilute that out considerably at the very end of the process. And then you're loading it on the flow cell, which if anyone watching doesn't know about the min ion we're talking about, definitely Google it, because it's like a little flash drive, basically, bigger than that, but still like an external hard drive, maybe. It's USB powered, at least, and that plugs into your computer, and so obviously that makes the portable aspect of nanopore sequencing. And they have other ones as well, bigger machines that can do human genomes and stuff. But um, the nanopore gets going, it gets running, monitoring that run during process. There's not much you can do at that point. You're mostly just sitting back and watching, but at least you can kind of see the pore count, the pore health we were talking about, and also just data accumulating. You can actually see like how many reads have been sequenced at this point, and how does that translate to number of bases and stuff. And, if you get 1 million reads in your run, like that's a really good run. In theory, and on paper though, it's the fungals are capable of even more than that. But that's kind of in a nutshell how the sequencing part of it goes. And also keep in mind that like we're talking about these are multiplexed, so after that PCR step, you're working with a single tube that has all of your samples combined. So at least that makes it a bit easier when you're doing the library prep. You're just working with a single tube at that point. But then you start getting into the bioinformatics and all the data analysis side of things. And at that point, some of it there, you do have to do a lot by hand, kind of like a lot of command line. You're typing in a computer terminal, kind of. There's no like graphical interface for these software programs. But you out of the sequencer you get this like raw data. Actually, in fact, it's like similar to Sanger trace file, where it hasn't even been interpreted into ACGs and Ts, or base called as so the first step is taking those raw trace files and base calling them into at least somewhat readable gene sequences. Because what the nanopore is doing, as the DNA strand passes through the pore, it's measuring electrical voltage current, and basically like depending on what base nucleotide is going through the pore, it'll change that voltage ever so slightly. So you can actually discern whether it's an A, C, G, or T based on the fluctuations. And they call it a squiggle, because if you look at it, it looks like squiggle, like a not quite like a cardiogram. It's more like steps and plateaus versus like peaks and stuff. But still, you base call it into your sequences, and then it goes through the series of steps of quality filters, and kind of manipulation of the data to demultiplexing is a key step we talked about, separating those samples back out based on the unique tags that we put on those molecules. We have steps for other quality things, like there are these things called chimeras, which are like if you had two different ITS sequences that were accidentally or mistakenly merged together. That can be like really confusing data to look through. So like removing those, generating those consensus sequences is the end goal. So we're piling up many reads of the same gene sequence, in the hopes of getting a single high quality sequence out of that pipeline per sample. But like Scott said, there is kind of an expectation that for some or most samples, you will have like contamination or crosstalk. And crosstalk isn't exactly unique to Nanopore, and different things can cause it. But basically, it's just like misidentified samples. Like they're put into the wrong sample bucket, the reads, due to some reason. And so sometimes you'll get multiple sequences, consensus sequences per sample. And part of the validator's job, sometimes it's like very easy to tell like what is your target, or your the specimen you're going after. And if you have a polypore and it's coming back as like an agaricus, like you're like, all right, well, this is clearly the wrong sample, and we can just throw that out. But yeah, like in the end, you end up with the sequences, and then that all gets uploaded to MycoMap, which is Stephen Russell's website that he has been developing for years now. And MycoMap does a whole lot of heavy lifting in terms of sorting through and really housing like all this metadata, and facilitating like tracking which sequences have been validated and which have to be validated. And it links out to iNaturalist. It'll update iNaturalist. Scott's definitely the person who knows the most. Why don't you actually take it over, Scott?
So yeah, this is our spreadsheet, and something that's worth highlighting is under this sequence status column, we have like whether or not it's passed or failed. So you'll notice a lot of fails. So yeah, that's.
So now this is all stuff that you've run through. So there's another list of stuff that doesn't have a good or failed yet.
Yep, there's two lists. Yep. One list is like just the initial sample collection. That's the list that we record going into the tubes. And then this is a list that we generate after the, all these steps that we just talked about.
All right, so this is run 12, and I'm going to go ahead and steal one of Alan's. So I've got the INAT number here. Here, I'll just. So I can pull up the INAT, and then I can search for the record within the project. Oh, and that one is not in the project. Let's try another. So that one was likely a fail. Sorry, Sarah. Watch, I'm going to pull up like 5, and they're all going to be fails.
Hey, that's good though, because the audience will definitely see.
Yeah, this is part of the deal.
Okay, so great. We have this Anasabaceae.
I was going to say, not that this was planned for Kyle at all.
Wow, let's see this. I'm in now. I'm awake.
Yeah, so all right, so we have the record here in MycoMap, and there's a little sequence box, and there are 2 sequences attached to this record. So on this page, we have the consensus sequence that's provided, as well as like trace files, the reads in consensus, just like some basic information. And on the right-hand side, there are the blast results, which I'll open. And when you import your file to MycoMap, it automatically does a blast search. And not only does it pull from GenBank, but it also does a localized blast, which pulls sequence data from iNaturalist, Mushroom Observer, what else? Unite,
Bold.
a couple, Bold, a couple people's own databases. Okay, and so this one actually, this one actually might not be Anasabaceae. So okay, we have 2 sequences. One of them is matching Amanita brunescens var pallida. I already know that this record does not go to that, so I'm going to trash that sequence.
So this is where you guys were talking about you might have multiple sequences potentially.
Right. So likely somewhere else on this plate, there was probably an Amanita brunescens var pallida.
Right.
And, you know, maybe Zach could speak to this, but for whatever reason, there was crosstalk, and the Amanita sequence likely got associated with the correct record, but it also got associated with this record.
Gotcha.
But okay, so now I have this other one.
And that's got to do with the primers, right? I have to assume.
Yeah, I was gonna say, so there could be multiple things, like crosstalk, I said, can be both like an artifact of the data side of things. It also could be what's happening in, like with Illumina. It's much more common, like in the sequencing run, you have DNA molecules that are like hopping. They call it index hopping. And but also like another common thing is like kind of one of the beauties of Nanopore is that it can all still work with contamination. Like we kind of almost expect contamination is going to happen. So like if there's a little bit of one sample, the previous sample on your stuck on your forceps, like we're cleaning the forceps in between one, but it's not perfect all the time. And so like something like that, that's not going to ruin the sample. It might if you were doing Sanger sequencing or something. But one other quick thing I wanted to mention, talking about multiple sequences, something that Nanopore again can uniquely handle very well is think about the cases where you actually have two legitimate fungi ITS sequences in a single sample. Like if there's a parasitic or symbiotic fungi, like hypomycetes is a common example. Like you actually would expect to get two ITS sequences of the host and the parasitic one.
You would expect, but for me so far, I only get the parasitic one for some reason.
Oh, that's interesting.
I don't know why that is.
It can be hit or miss like that, where sometimes you'll get what the thing is parasitizing, sometimes you'll get the parasite itself, and sometimes you'll get both. But yeah,
Nanopore, there's the potential to get both. Whereas with Sanger, there's no that doesn't.
You're just gonna get a composite of the two, and it's gonna be useless data.
Useless.
Yeah. So here, and I'm gonna assume this is right because actually, now that I see sequence results, I'm like, oh, that kind of does look like a Cordonarius. So this top section, NCBI blast results. So this is, if you were to do a blast of the sequence within GenBank, this is the top 10, and I can show more. This is going to be the top 10 matches you're going to get if you were to just blast it in GenBank. And so the top match is what Stephen's calling Cordonarius species IN61. But then down here is the mycomap or the mycoblast results. So this is pulling stuff from iNaturalist, Mushroom Observer, and other places like Unite, like I mentioned. So I'm just going to go ahead and open a couple of these that are within 99% range and compare the morphology. And okay, so this is one of my pet peeves as far as like photos. There's a single photo, you can't see the gills.
Yep.
I'm just going to ignore that one. Okay, so this one is a bit better.
Yep.
And it's looking definitely like a Cordonarius. I would say it does resemble, Sarah's observation here. And then we have another, again, by John Plischke. Just for the heck of it, I'll open a couple more. Great.
Yeah, you're in the ballpark there.
Yep.
All right. So now I've confirmed that's what it is. But okay, so you'll notice here, I'll just use this one as an example. So under identity here, you have two numbers, and this represents the percent match. So if you were to blast this in GenBank, GenBank would provide an unadjusted percent identity match, which is 98.77. But then MycoMap does this thing where it accounts for certain, how do I put it? I guess idiosyncrasies or more technically repeating homopolymer regions and IUPAC codes. And so it has this adjusted identity of 99.82. So you'll notice in this alignment, there's a couple green positions. So like this one here, we have a T and they have a Y.
Right.
And Y isn't A, C, T, or G. So why is there a Y in their sequence? So Y represents that in this nucleotide position, there could be a T or a C. And a T or a C is the most common IUPAC, or Y is the most common IUPAC code you'll use, because, well, so it represents like the differences within the ITS within the organism. So there's what, like 100 copies of the ITS, Zach, roughly?
It's definitely variable. It could be 20, it could be 50, it could be 100.
Okay. And not every ITS is the same within a single organism. Some of them, in this case, at least in the sequence we're comparing it to, have a T in this position, and some of them have a C in this position. Our consensus read spat out a T, and so MycoMap is going to assign less weight to this difference because there's a Y here. And then down here, this red G indicates what would be considered like a real nucleotide difference. But I can't assume that it's actually a real difference unless I go in to the trace file, which I can find here. And I should be able to do this kind of quickly. So I don't want to bore people. Oh, shit.
We're lucky we have Scott because he has this memorized.
He does. He is the look. He even knew.
Good old ag tacata.
Yeah, he just sped it right in there. Didn't
Just
even.
ag tacata.
Okay, so what I did, basically that agat kata, AGGATCATTA, represents a motif at the beginning of the ITS sequence that we can see right here. And this tells me that when I'm looking at what's called a FAST2 file, that all these reads are in the forward position. And I actually got pretty lucky that they're all in the right direction. Usually it'll be split about 50-50. Some of them will be reverse complemented. Some of them will be in the forward direction, and I'll have to correct that. But great. So all these are in the forward direction. So just for the hell of it, I'm going to go to where there was a Y in theirs. And actually, so that's this position. We have T's pretty much all the way down.
Yeah.
I can do a quick hover. So we have 84% T's, 10% C's. If it were closer to like a 50-50 distribution, I would probably add the IUPAC code Y. Let's skip to this real difference, potentially. Okay, so here, we have a pretty equal distribution of C's and G's, which Eugene is calling it an S. I actually forget if that's the right thing. Sometimes it gives me the wrong thing. Okay, so G or C, S. So I can go back. Just make sure, and I can modify that G to be an S. And then do that quirk of MycoMap. But in the meantime, I can go ahead and so this is being called Coordinaria species IN. I forgot the number, but IN something, 61. I'll add that to the provisional species name field. And this way you can, I can like click this and go to observations with this field and value, and I can see every observation on INAT with that name in the provisional species name field, which is great. And then I'll go here, Cordonarius. And if I wanted to be a little more specific, I could figure out what section this is based on close matches with a name. It's taking a while. And I could say, oh, it's in Cordonarius. Oh, shit. Okay, MycoMap might be having an issue.
Hey, it is midnight.
It is midnight, yeah. Stuff happens at midnight on MycoMap. But anyway, I can also go ahead and oh shit, yeah. From here, I can usually hit import to INAT, and then it'll populate all the relevant observation fields with the ITS, the MycoMap blast result. Here, I'm just gonna make a note to.
Yeah, Steven has really like custom made MycoMap to be like
everything GenBank should have been.
Well, yeah, I mean, they're different things, but like it really is, you know, perfect for exactly what he's doing and everything too. Like especially with the integration with INAT and like Scott said, like the database in MycoMap for ITS sequences now dwarfs like what is in GenBank, and so like that's just super valuable as well.
GenBank is like the Medicare, and then MycoMaps is like that bomb ass supplemental insurance you get where you get to go to all the best doctors and get all the good information.
Yeah, maybe that's a good way to put it. But you know, I don't like to harp on GenBank too hard because like, man, like someone has to do it, and it is like you know, they host terabytes of genetic data for free for all sorts of stuff. But
yeah.
What can you do?
I'd love it if they hired somebody to touch it up a little bit.
I also knew some people that worked at NCBI on like GenBank stuff, and can't trash talk them too much.
No, not in public, at least. Yes.
No.
Sure. So here's I what I hope everybody gets out of this is a long journey from the woods to the internet. To your INAT account with, you know, this pretty little cluster of, you know, 4 different letters repeated endlessly. And what does it mean? Sometimes it means Scott can pretty authoritatively say, this is the genus. Sometimes you can get, quite often you can get to species. Sometimes you go, wow, GenBank's never seen this before. This is, you know, potentially something brand new. We don't know, might not be. It can go a lot of different ways. It's very exciting. Sometimes it's like, hey, we got countless matches, 100% matches. We got holotype matches. We got, you know, a plethora of validating data. Other times it's you were the first person to ever upload anything in your state. Sometimes it's this doesn't exist anywhere on GenBank, period. Can go a whole lot of different ways. And it's all fun and interesting.
Now would maybe be a good time to plug if you are interested in this whole process. The 3 of us will be teaching a DNA workshop with from the farm fungi outside of
Gary Hefferly. Oh, cool, man. You guys get to hang out with Gary. It's my buddy.
Yeah, Gary's great. Such a nice guy. Has a beautiful house that he's turning into like this mushroom compound.
Are you gonna go hang out in one of his yurts, guys?
I hope so. I don't know. Like he was telling me about his plans for the yurts, which sounds awesome. Like truly making it like this mushroom mecca. Because he's already doing classes. And he's done a lot of classes in the past. Well, I visited him earlier this year, and he had the idea of us doing a workshop once he heard like what we were doing at OMDL. So we were like, hell yeah, let's do it. And so we just recently like the tickets for that went live. It's going to be over Labor Day weekend. It's like the last weekend of August 31st and September 1st. It's a 1 day thing, but we have 2 days that we're doing it. And the tickets will be limited to like 8 to 12 people per day. So it is kind of a limited number of seats, but um, I think we can get the link to you to put in the description. So if people want to get hands on and learn more intricately, like basically everything we talked about, really, DNA extraction, PCR, running gels, and then getting into the sequence validation stuff in more detail. If you're interested in that, we'd love to have as many students as we can to teach, because we're all really passionate about this.
End of August. It's 1 day, but you're doing it for 2 different days, 8 to 12 people. Oh, we gonna book that up. I mean, that's gonna book up, dude. You don't even gotta worry about that. That's gonna be fully booked.
Yeah.
Like, that'll probably be fully booked in a few days after this airs, for sure. All the PCR amplification stuff, gels, you guys are going over Nanopore and Sanger, like giving people a lowdown on Sanger, or are we
Yeah,
strictly Nanopore?
we're gonna try and cover both, in kind of, because like I said, the methods for Sanger, they are transferable to Nanopore, and it's also much more accessible if someone wanted to like get their foot in the door with this kind of stuff. Like Sanger makes far more sense to get started. So we're gonna try and like cover both and really make it worthwhile for people. And we're also going to we're trying to figure out if we can do a live run of Nanopore, maybe like loading it and running it. The problem is, something I didn't mention is the Nanopore run takes like 24 hours, sometimes more, to run. So like figuring out the logistics and timing of that can be tricky. In addition to like the extractions and PCR that Kyle mentioned also take a couple hours to run. So we're going to kind of like do some hands-on stuff, and then while that's running, we'll do some lecture kind of like PowerPoint stuff to cover more things, and then go back and forth with more hands-on stuff. That's our idea.
You just got to get there 2 days early, and you have 2 different flow cells you run, and you run them back to back. Zach, you bring yours, Kyle brings his, and you get them perfectly timed so that one ends on one day and one ends on the other. I'm joking, but I'm also not. That's how you do it. You could do it.
That's it. Like I've taught workshops before. We didn't necessarily do live sequencing, but like, yeah, you got to do some like TV magic behind the scenes to have some stuff prepared.
And it would be cool just to, I mean, I thought it was cool just to watch it run. It's also cool when it's done and he starts talking about the data and, here's how much data we got. It was really cool to see the pore count go down or magically up for a minute. And just in general, any little taste of that, I think, would be great for people to see. They'll like that. But man, if you guys are interested, you get to go up to Gary's amazing little piece of property up there and learn from. Oh yeah, it's crazy.
He has a gorgeous view. He's like in the foothills of the Rockies there, a little south of Denver, and it's just beautiful.
Yeah. So Kyle, Zach, and Scott are going to be there. They're going to be teaching everybody all about this stuff. So if you listen to this and you said, "Oh, that sounds cool. I like all that. I'm about that life." You can be about that life. You just cruise on up to Denver. I mean, there's a lot of people in Denver right now that might be all about this. So I'm thinking you guys will be booked up real fast. So get you one of them tickets. Get up there. You don't know when they're going to be back again. This is what I tell everybody. If there's a cool mushroom thing happening in your neck of the woods, go. What's your problem? Just go. And somebody go, I don't know. I'm an hour away. I don't know if I can go to the Ohio Mushroom Festival this year. Don't. Where are you gonna go, dude? Where are you gonna go instead? You're gonna go to the Alabama Mushroom Festival? That's farther. You can go there. I'm not saying don't go there. This one's 1 hour away. Go to the shit that's by you. When people from other places are coming to you, go to those things. It's not rocket science. Anyway, so we'll get the links up for that. I imagine Gary will have it on his site as well. Yep. That's gonna be a good time. You guys gotta do a little IG live stream here and there. You gotta, show people off.
Oh yeah, Kyle will be live a couple times.
I also maybe want to stress like the role that other people played in all this coming together. Stephen Russell started this whole thing, and like for me, I got into like the whole DNA sequencing thing partially because of Alan, and definitely because I found that psilocybe. But I got into it and I was casually getting stuff sequenced, and then that led to developing the skill set to be able to analyze DNA sequences, which landed me a job with Fundus. And, Hart Singer, the sequencer for Fundus, one of the hardest working individuals I know in this community, he got into the Nanopore. And so he got the Fundus team on Nanopore sequencing like 1 or 2 months before Kyle started up, which allowed me to get that practice in and allowed us to just, get the ball rolling with like sequence analysis for all this. And then I was able to train Jessica and Sarah and Alan and, all our validators. But, all these things are like all these different like labs. All their work is so tightly interlaced. Like the work that Steven does, the work that Kyle does helps create reference sequences for Fundus, because we're doing stuff, Kyle sequences a bunch of stuff from Washington and Oregon and a little bit of California. And, that helps me as a Fundus validator, because then we're generating sequences that I can use as reference for Fundus, and vice versa. Fundus does it for us. And, there's a lot of niches you can occupy in this space, and there's a lot of bottlenecks. And like you said earlier, what was it? Like many hands makes light work.
Yeah.
So if you're interested in maybe not necessarily doing the whole thing, but just like occupying one of these niches, we definitely need more people in this space, and more people making more collections, developing relationships with universities. And that's something that wasn't really touched on, but after these things get sequenced and we have the sequence data, it's really valuable for someone in the future to study that collection. And so if you can get it accessioned into a fungarium at a university, or even establish your own, then someone can request a loan of that specimen in the future if they're studying that genus. And but I do want to emphasize the university thing, because building the bridge between the academics and the community mycologists is extremely important. And I've been going to UF a lot lately, and there's just like so much work to be done in their fungarium, and I've been trying my best to help out there. But if we really want to like push mycology forward, this is necessary work.
Yeah,
it is. It's a lot of work. It's very tedious for 4, 5, 6 people to do. If you got 4,000 people coast to coast doing it, it gets a lot easier, get a lot more stuff done. So yeah. But for sure, at a minimum, like Kyle and I said on an episode a long time ago, like the old bottleneck was the sequencing. The new bottleneck now is the specimens. So yeah, get out there, find mushrooms. Just go find some mushrooms, take some pictures of them, dry them, send them to these guys. Let them take it from there.
And good point bringing Stephen Russell up, Scott. Yeah, big shout out to Stephen Russell. Completely inspired by that guy. His protocol being free, accessible, amazing. He never had to do any of that, and he did, and he took me under his wing. So great thanks to him. Alan also inspired me in DNA, and lastly, Sigrid Jacobs. She has an awesome 4-part series on YouTube for Sanger sequencing. So if you are getting started into Sanger sequencing, I would just say watch that 4-part series, watch it twice, and you can feel confident. Great thing. And then lastly, again, shout out to all these all these guys on the team: Jessica, Sarah, Scott, Zach, Alan, Mandie, Grant,
Josh Birkebak
Birkebak.
has
Oh,
been instrumental in a lot, like helping us with publishing papers and
yeah. And another thing, we are a free sequencing service, but I also didn't mention, but everybody on this team, nobody gets a paycheck. So they're all doing it out of their pocket, their passion, their will. There's no travel pay. We link up, we drive to each other's houses. It's literally a group of folks that are extremely passionate about mycology and about this new awesome thing we're in with DNA. And yeah, I think that's super important. Not saying that we won't get paid one day. We are a nonprofit and we are looking for funds, but to let everybody know, all this work is 100% done for free. Everybody's time, everybody's management. We have families, and things come up, and everybody does their best. So it's important as well for the folks to know.
And Kyle does have an open GoFundMe that goes to the expensive reagents when we need to place that 12 fungal order. It's straight from that GoFundMe.
I mean, flongals don't buy themselves, reagents don't buy themselves, primers don't buy themselves. Yep, it all these free sequences still cost money.
For sure.
Yeah, well, so we'll set up a link for the GoFundMe as well. And Kyle already knows this, but anytime you're really looking for a push to raise some funds to get some more stuff going, let me know. I can definitely make sure that it gets mentioned on a weekly episode. We'll take care of you. We're gonna keep, got to take care of the Ohio boys. Got to keep all this going. So, all right, guys. Well, this was great. For the people who are just getting into walking around the woods and finding mushrooms, I think this gives you, a little bit of purpose behind some of that. And like I said, a lot of my audience already got a dehydrator, so they're ready to go. You just gotta go to the Dollar Tree and get you some $1 Ziploc bags and a Sharpie, and you're good to go. Just start getting these samples in. All right, guys. Thank you so much for being here. Really appreciate you guys taking time out of your day to talk to everybody about what you guys do. And also, Kyle just said it, but I think it's worth repeating. There are a lot of really amazing people who have been passionate about mushrooms for a long time. There are people who are just finding that passion, a week ago, a year ago, a month ago, whatever it is. And all those little hyphal branches are finding the old rhizomorphic growth, and it's all coming together. And when that happens and it starts building and building, that's when the really exciting stuff happens. And it's so cool to know you guys and to see it happen. It's phenomenal to get sequences back. I'm probably going my iNet right now. If Sigrid probably got another few in for me.
Probably did.
That's accurate. Yeah.
Probably. Anyway, all right. Thank you guys. And we will do this again soon.
Yeah, thank you so much.
All right, guys. That was OMDL, Ohio Mushroom DNA Lab, led by Kyle Canan, trusty sidekick Scott Ostuni, Zach Geurin, and then all the other people. Let me make sure I'm not gonna forget anybody here. We got Stephen Russell, the founding father, the one who really got the ball rolling with Nanopore min-ion sequencing. He did a lot of the heavy lifting on that, a lot of trial and error, a lot of figuring stuff out. And now his MycoMaps, is working so well with GenBank and really starting to crunch out some of this data in a very useful way. Joshua Birkebak, he has helped the OMDL guys publish some great papers on novel species and what they've been up to recently. Alan Rockefeller, as always, for crucial identification, for microscopy on some of the papers, all that good stuff. He's always helping people out. And then a couple local gals that I know and get to hang out with from time to time, Jessica Williams and Sarah Culliton. Can't forget Mandie Quark, Grant Cain, Sigrid Jakob, and Logan Borosch. Sorry, Logan. I'm sure I just murdered your last name. Anyway, it's quite a team. They do a lot of work. I really encourage you guys, go drop a $5, $10, $25 donation, especially if you've ever submitted samples to the lab to be sequenced. Show your appreciation. Little reciprocity. I've been talking recently about the lack of reciprocity in the community. That's been a little disheartening for me and countless other people I've been talking to. I don't think it's the mushroom community. I think it's the community at large. We're getting just a little self-centered. So, just take a moment and appreciate the people who are doing cool things for you. We'll have links to Kyle's GoFundMe. I'll have links for the Joan Maloof book. What else? I'm gonna put in a link for how to observe mushrooms methodically on iNaturalist. We're gonna include a link to Sigrid Jakob's PCR videos, and then all of Kyle's links. So you guys will, if anything interests you and you want to learn more, should have all the links in the description for you guys. So anyway, also want to let you guys know. The Mycotrex Mexico trip is, I think, like a month away, and we got a lot going on, a lot of things to deal with there. My kids are home now. I'm being pulled pretty thin these days. I just want to give everybody a heads up. We might not be doing fresh new content every Monday. I might have no episodes. I might have do an old rerun that I might have some time to do some editing on. But basically, between now and at least after I get back from Mexico, or possibly into August, we're going to be running a little bit light in order to create great content and to evolve as a podcast host. I gotta get out there. I got to learn things. I got to explore. I got to meet people. And so this is part of that evolution. So during the summer, man, it's like prime time for growth, both of mushrooms and of my mycology interests. So this is not, I'm not stepping away. I'm going deeper, but in order to do that, there's a point where I am literally one guy. And after careful consideration, someone I was talking to recently was like, "Man, just you don't got to do an episode every week. Like, if you need to get out and do some of these other things, go do them. People will understand. It's okay." Well, no, I've just been like so wanting to prove to everybody that I care about this community, that I want to be here for you guys, that I want to be here every Monday night for Myco Monday nights. And so I've been really resistant to that, but I think it's important, and I'm taking that advice. So just know this. If I can get an episode up, I will in the next month and a half. If I can't, it's only because I'm out trying to get this trip together, trying to go out and forage, learn more about mushrooms. So we got more to talk about. So anyway, hope you guys understand. If you get really mad, you can send me a DM and ball me out in the DM. That's fine. So, guys, I can't wait to be back with another fresh episode. Cannot wait to be back with all sorts of content from the Mexico trip, from MycoFest, from Ohio Mushroom Festival, and maybe we'll see a couple other festivals I'm hoping to get to. We're going to get out there and do some fun stuff this summer, and bring you all sorts of content, let you know what it's all about, hopefully inspire you guys to go on some of these trips, go to some of these events. I will keep you guys updated on the events I'm going to. So if you want to make a trip, come hang out. I'd love to hang out with you guys. Anyway, until next time, go grow some mushrooms.